All right, so our first talk is going to be about rust disease of Blackberry and cane disease management update from Georgia, and it's going to be Dr. Jonathan Oliver, so yeah.
All right, thank you, Aaron. So yes, my name is Jonathan Oliver. I'm the fruit pathologist and extension specialist at the University of Georgia, based in Tifton. And I'm going to talk primarily about rust diseases on Blackberry today, but also kind of give you an overview of a caneberry disease management update. So first, one of my responsibilities at UGA is to be involved with the diagnostician and make recommendations on samples that come into our diagnostic lab. So I usually like to start talks by talking about some of the diseases we saw this past year. And then I will talk primarily, like I said, about rust diseases. And I'll end by talking about the seasonal spray schedule for caneberries and some additional resources that can help you develop your spray program. So first, the diagnostic summary. In 2024, we diagnosed, the diagnostic clinic diagnosed 11 different issues on Blackberries. This is a pretty low amount of diseases diagnosed relative to previous years. 2023 was also pretty low and had 15 issues, but primarily we see a lot of cane disease issues as well as some leaf and fruit disease issues. So specifically, I was going to mention, mostly the difference between these 2 years is we saw less cane blight on samples submitted to the diagnostic clinic this year and less root diseases in 2024 relative to 2023. But we did see orange cane blotch, this algal disease that I'll have a slide on a little bit later, in several samples. We did see some cane dieback that we initially thought was being caused by the cane blight pathogen, but instead what we found was some Botryosphaeria-related species causing that dieback. We also saw cane and leaf rust, which again will be a major topic of today's talk. So moving into rust diseases, there's two major rust diseases that impact blackberries in the southeast, and recognizing which one you have can be really important. So I'm going to talk about recognizing cane and leaf rust versus orange rust, and then I'll talk about the management of those rust diseases after that. So these two major rust diseases of blackberry are orange rust here and cane and leaf rust on the other side. And they appear to be superficially similar. They're both rust diseases, but they require very different management practices due to the biology of the fungal organisms that cause them. And so it is important to know which one you have, because a rust isn't always a rust, isn't always a rust. They're not the same, but they do appear superficially similar in some ways. They're both diseases caused by fungi that are basidiomycete fungi. They both negatively impact plant health and result in yield losses. Sometimes when I get sent a picture from a grower or an agent, they'll ask, is this the bad rust or the good rust? There's not really a good rust to have, but there's one that's definitely worse to have than the other. And so we'll talk about what that means, but because they both actually negatively impact plant health. They produce orange and yellow pustules on the leaves, regardless of which rust you have, but the pustules are one way you can kind of tell apart these two different diseases. And most importantly, knowing which one you have is important because the control recommendations are quite different. All right, so the disease orange rust is actually caused by a couple different fungal species, and these fungal species infect the plant systemically. And this is important, and this is a very important distinction, because since the plant gets infected systemically, once the plant is infected, you can't just prune this disease out, or you can't, some people have asked, can I just mow these plants down, and will the new canes still have rust? If it's orange rust, they will, because the plant's been affected systemically. The infections cause dramatic reductions in the vegetative growth and the production of fruit. And both of these, you don't really need to know which of these two species of fungi you have, because both of these cause orange rust, and their symptoms are pretty identical. But the symptoms first appear on the new growth in the spring, and so the shoots that emerge will appear kind of spindly, and the unfolding leaves will appear stunted or distorted or pale green. They just don't look quite right. And about 3 weeks after those leaves unfold, you'll start to see these blister-like fungal structures here. These are acea that produce acea spores, and they'll develop on the undersurface or the margins of these leaves, and they'll actually cause the leaf margins often to kind of distort or curve, and then they will grow powdery and bright orange as the spores are eventually being shed. So by early summer, on those plants that have orange rust, you may not notice too much of a problem. You may only see issues on a few of those canes. And so that may deceive you, but the entire plant, when it's infected with orange rust, should be thought of as systemically infected for the rest of its life. The roots of the infected plants produce a proliferation of these spindly shoots, and the floricanes will ultimately not have flowers. And of course, without flowers, you don't get any fruit, and so these plants, once they're infected with orange rust, are basically useless from a commercial standpoint, and that's why we recommend their removal. But you can see the leaf margins here that are distorted by this, and underneath, on the underside of the leaf, you see all these pustules present, and these are all symptoms of orange rust. So the acaeospores are what are actually produced from these orange pustules, and if you look at them under the microscope, they're these roundish spores, and I'll show you the spores of the other type of rust pathogen later, and they look a little different, of course, but these are the spores that are produced, and they're spread from plant to plant by wind and rain splash in the late spring, and they infect new leaves whenever there's free moisture. And of course, in the southeast, we have lots of rain, so we have plenty of free moisture often for these diseases to spread and infect. These infections give rise to additional types of fungal spores, and which give rise to additional fungal spores, which ultimately cause the systemic infection of both the crown and the roots, and then it definitely is affected for the rest of that plant's life. It doesn't necessarily kill the plant, though, and the plants can hang around there for a long time being infected, which is actually, if you think about it, more of a problem because they can continue to spread the pathogen to other nearby plants. So cultural controls are really critical for managing orange rust. So first, you don't want to have it in your field or adjacent to your field in the first place. So how can you prevent problems initially with orange rust? It's really important to ensure that the plants are free of the disease. And so one way you can do that is to use plants produced by using tissue culture if possible. Most of the issues that we've seen with orange rust, when I get sent samples or pictures of something that turns out to be orange rust, at least half the time since I've been at UGA, it's been because those plants were propagated from a neighbor's field, or it was somebody who thought the wild blackberries growing near their house were really nice and propagated some of those and had some of those growing. And so often it is a disease that is coming in from plants that are already infected. It also is a problem, though, of wild blackberries, and so if there are wild blackberries within about a quarter mile of your planting, these spores can move into your commercial planting. And so if you can eradicate the wild blackberries, that can reduce problems with orange rust. Once your planting is established, there's less you can do. You can thoroughly inspect the new canes. Again, when they're coming up, they'll look a little distorted and odd. When they're 12 to 18 inches high is kind of the key time for inspecting those canes. And then if you find any orange rust, those plants should be removed and destroyed as soon as possible. Because again, they're not going to be productive, and they're going to serve as a source of continued spread of the orange rust pathogen. There are many cultivars that have some resistance to orange rust, and so I've listed some of those here, but Primark Traveler and Primark Freedom are considered susceptible, so you can get, and there are some other commercial cultivars, especially some of the older cultivars that are still grown that are susceptible to orange rust. So as far as controlling orange rust through chemical management, chemical controls are really not effective once the plants are systemically infected. Now that being said, the group 3 fungicides, the DMIs, and the group 11 fungicides, the strobilurins, are rated as excellent for their control of orange rust, but that's more or less an excellence in their prevention of new infections from occurring. So the plants that already have orange rust, using these fungicides is not going to eliminate the disease. Really, once you find orange rust, that plant should be removed, and these applications help prevent infection of nearby plants that are not yet infected. And that's a very important distinction between these two types of rust. So cane and leaf rust is the other type of rust. So orange rust is what some people call the bad rust. Cane and leaf rust isn't a good rust for sure. It definitely causes significant economic losses in blackberry production, but it's not the death sentence for the production of the plant like orange rust essentially is. So it's caused by a different fungal species that does not infect the plant systemically. And this fungus, unlike orange rust, produces pustules both on the leaves and also the canes. That's the name cane and leaf rust. So orange rust does not produce pustules on the canes, but cane and leaf rust does. And so that's another way you can tell them apart. So the symptoms for this rust are usually first spotted on the floricanes. You'll have these lemon yellow pustules that develop on the bark of the floricanes. and they will split the bark. And then the infected floricanes that have those pustules, the pathogen does negatively impact those canes, and so they're going to be stunted and delayed in development and not as productive. In contrast, though, like I mentioned, to the cane and leaf rust pathogen, the orange rust fungus does not form pustules on canes, so that is a distinction. So in the spring, on the leaves, and this is what we usually see first in South Georgia, we'll see leaves that are hanging around since last season, and some of the earliest leaves will have these small pustules. These are called uridinea, fungal structures that produce uridinea spores, and ultimately, these spores spread from plant to plant, and you can get severe enough infection that the plant can defoliate as a result of cane and leaf rust. And if it defoliates early, the plant's not going to have stored up enough reserves to make it through the winter, and so you can have more winter injury, and those plants will be less productive next season. So it does have an impact. The pustules, though, in cane and leaf rust on the leaves are different than the orange rust pustules because they do not distort the leaf margin. So again, the orange rust, often the leaf margins will curl. Cane and leaf rust, they don't. So another way to tell them apart. Color-wise, the pustules from cane and leaf rust are a little more lemon yellow in color than the orange rust pustules, but I've never really found color to be a very reliable way to tell these apart because as the pustules age, they kind of get a little darker, and it's very subjective of what is, light orange and bright orange and dark orange. So that's not really recommended to tell them apart, but there is a little bit of difference in terms of color. So in terms of the disease cycle, the fungus overwinters as mycelium or as latent uridinea, and in the spring, these uridinea on the floricanes release those spores and they infect nearby leaves. And you can see the spores here, they're a little less round and they're more likely to be pointed or a little more triangular than the spores from the orange rust fungus. Wet conditions, which again we have plenty of, favor disease development. And then in the summer and the fall, you have the other spore stages which infect adjacent primocane leaves. So how is cane and leaf rust managed? Well, it involves culturally removing the floricanes after harvest. Generally in blackberry production, the fact that the canes are around for 2 years is a problem from a disease management standpoint because basically if those canes get infected as a primocane and you have to keep those same canes for next season to produce fruit, those canes will already be infected with pathogens, and whenever you have floricanes and primocanes there at the same time, you can have transfer of these pathogens from one to the other. And so from a pathology standpoint, once the fruit has been harvested off those floricanes, they should be removed as soon as possible to limit the amount of time transfer of pathogens can occur from one to another type of cane. Chemical controls are the same, actually, as I mentioned for orange rust. So the strobilurins, the group 11, and the DMI, the group 3 fungicides are recommended. And fungicides for cane and leaf rust should be applied pretty early in the season. We see this one of the first diseases we see in South Georgia every year. So applications at green tip and prior to bloom are important. In the Pacific Northwest, applications early in the season are usually enough until later in the season after the floricanes have been removed. If you scout and find this disease, our growers often have to make applications straight through from early season, through the harvest period and then after harvest. Because most of our fields have a history of cane and leaf rust, and so they need those additional applications because it's a major issue. Again, this is the same table I showed earlier with orange rust. Those same fungicides are recommended, but in this case they do provide satisfactory control rather than just kind of a preventative of new infections occurring, like in the case of orange rust. So a quick side-by-side of these two rust diseases and the different things I've said. So both of these are significant blackberry diseases. There's not really a good rust to have, but the fact that orange rust is a systemic fungal disease makes it the worst one to have because it makes it more challenging to control. And cane and leaf rust, by contrast, is non-systemic. Cane and leaf rust, the pustules are a little more lemon yellow, and they're found on both canes and leaves. But for orange rust, these bright orange pustules are only found on the leaves. The leaf pustules with cane and leaf rust don't distort the margins of the leaves, but they do distort the leaf margins in the case of orange rust. Floricane removal after harvest is recommended as a cultural practice for cane and leaf rust management, but for orange rust, a complete destruction of the infected plant as soon as possible is necessary. Sanitation and fungicide sprays typically can provide satisfactory control of cane and leaf rust, but fungicides only reduce new infections of orange rust, and infected plants need to be destroyed as soon as possible. There are some other diseases that look like rust on blackberry. There is a yellow rust, but typically that's only found in other locations, not in the southeast, on raspberries and some black raspberries. But there's also orange cane blotch, which I mentioned before is an algal disease that superficially at least it can look a little like a rust. The blotches left behind caused by the algae are orange, and they do form sporangiophores, which give those lesions, those blotches, kind of a felty appearance. So this disease is also called orange felt disease. But it can easily be told apart from the rust diseases. If you need help, your county agent or the diagnostic lab can easily tell these different diseases apart. But anyway, it is a look-alike that sometimes gets confused with rust. So now I want to talk about the seasonal spray schedule, including the major blackberry diseases and the key spray dates and timings for managing those diseases. So we have all of these diseases in the southeast on blackberries. Anthracnose is a little less common in Georgia, but it seems to be more common in other locations in the southeast, including out in Arkansas. Cane blight is a major problem in Georgia for sure, wiping out plantings and in Georgia as well as other parts of the southeast. It is a major issue when it gets going. Botrytis fruit rot is the major fruit rot that impacts blackberry production. Rosette or double blossom can be a significant disease, but a lot of cultivars that are grown commercially now have resistance to that disease. Pseudocercospora and Septoria leaf spot are important leaf spot diseases of blackberries in the southeast, but I believe the next speaker is going to talk on that topic, so I won't spend too much time there. And I talked about orange rust and cane and leaf rust and orange cane blotch a little bit. We also have problems with Phytophthora. So all of these diseases typically require in-season management. And how does that management look? Well, if we look at each of these major diseases that I've talked about and kind of when management interventions are necessary from the delayed dormant period as you go towards the after harvest period, you can see that our plants are vulnerable to these diseases at many times of the year or long periods throughout the growing season. And if you were managing all of these diseases, as some of our growers in the southeast have to deal with all of them in a given season, a spray schedule may get to be pretty intensive. And so this is just an example of a spray schedule. I'm not endorsing any of these particular products. This is just a list of potential sprays you might make, and I've color-coded those to show what they're potentially targeting at that time. So, this is purple and green because a spray of lime sulfur or sulfurics early in the season can reduce carryover of things like anthracnose or cane blight. Again, the early season sprays with something like a rally, tank mixed with Captan can be helpful against rusts. Botrytis, that major fruit rot I talked about, is targeted by sprays of switch, and again, rust still may be an issue, so sprays of rally along with switch, and something like Captan to protect the efficacy of switch against botrytis, which can develop resistance very easily, may be necessary throughout the period from bloom through harvest. Pristine is effective against multiple diseases here, so that's why I have it shaded in black. About late April in South Georgia through early August is the time period where the orange cane blotch algae is moving from floricanes to the new primocanes. And so applications of Prophyte targeting that disease during this time period can be very effective at reducing problems with orange cane blotch. Cane blight often is thought of as a wound pathogen, and of course blackberries are wounded often through pruning and other practices. And so anytime pruning is undertaken, applications of a broad spectrum fungicide like Pristine or Rally can be used to to prevent infection of those wounds with cane blight. And then after harvest, and this after harvest period can be extended in parts of the southeast, it's probably going to take multiple applications of DMI or other types of fungicides that are effective against leaf spots and rusts to manage those diseases. So that's kind of just an example of a spray schedule and what it might look like in conventional blackberry production in South Georgia, where we have almost all of these diseases on a regular basis. So some additional resources. You're probably aware of the Southeast Region Small Fruit Consortium's IPM guides that are available for caneberries. So I'm the editor of that guide. This is the 2024 guide. That's what's up currently. There have been a few minor changes that will come out soon in the 2025 guide. I'm actually waiting for one collaborator to get back to me so I can fix one little part of the guide, and then it'll be ready to go for the 2025 edition. And so, but no major changes in it from a pathology standpoint. I've highlighted a couple of those. So within that guide, there's a seasonal at-a-glance disease management options for caneberries. And if you, kind of like I showed before, look at the different developmental stages across the top of this chart, from delayed dormant all the way through after harvest, it has a list of those diseases that are significant at each of those time points, as well as a list of recommended fungicides within that guide for managing each of those diseases. I highlighted the changes. Again, not many changes in the 2025 edition. The fungicide also has been added for botrytis to that guide, and we've also put asterisks next to those options that are OMRI listed. So also is an OMRI listed fungicide for growers trying to grow organically, and there are some lime sulfurs and coppers that are OMRI listed, and so we've highlighted those a little better this year in the guide. And also in that guide, there is an efficacy chart that lists all of these major diseases and the efficacy of each respective fungicide that we would recommend in the Southeast against those particular diseases. We've added Sevia this year for its potential efficacy against leaf spots. It's a relatively newer label for Sevia for Blackberry. And we've also, like I mentioned, added Osso for botrytis as well. There is a My IPM app available, and that can be particularly useful in things related to the topic of today's talk, because it has lots of pictures of orange rust and cane and leaf rust and all of these different blackberry diseases that you can scroll through very easily in this app. The app's completely free through the App Store or Google Play, and once you find what disease you have, you can click on these links below, and it'll list the chemical controls and their relative efficacy. All the efficacy information found in the IPM guide is updated each year and put into this My IPM app. And with that, I'd be happy to take any questions you have if I still have time, which I think I do.
Thanks, John. For a few questions.
Any questions, Sarah?
Is Home Depot registered on Caneberry for botrytis? I wasn't at the meeting, you know, I had family issue, but we've done work with it.
Yeah.
It's been really great.
So it has worked for you?
Yeah.
We've done some work with it on blueberry, but I haven't looked at it on
Botrytis has
Blackberry.
looked really excellent.
Okay.
It's good to switch post-harvest, I must say. Yes.
Okay.
Yeah. It's really
Oh,
good.
I'd be interested in seeing that data.
So,
We
yeah.
we could potentially add that
Yeah,
to
I'll
the
send it
guide.
over to you.
Okay. Yeah, that's a good point. It has had some efficacy for blueberries
Yeah,
anyway.
it's looked good, I was surprised.
Yeah.
Only for botrytis, though?
We've tested it in other yeah, it looks great on mildew and apples and other things, believe it or not, but it's an alternative, though, and I mean, it looked really good post-harvest.
We'd love to add some more
Yeah.
organic options to the guide if we have data for them. So,
That's the only one I would, yeah.
okay. Yeah.
Like different cultivars, the
Uh-huh.
orange rust for us is worse than Napa variety.
Okay.
It's more severe. When it comes to the leaf and cane rust, have you saw any certain variety that's worse?
The varieties that we're growing seem to all get cane and leaf rust, so I can't say. I definitely have seen it in Ouachita for sure. That's one of the major cultivars we have, and I've seen it on Ouachita for sure.
Yeah, what we've seen is anything that holds its leaves a little more
Yeah.
seems to hold on to some of that knocking a little bit easier. You can find it in the
We can find it really early in the season.
in seedlings.
Yeah, really early. So yeah, I haven't. My observation is that seems to go to everything. So I'm not sure if anything's been done looking at that, but
Yeah, it seems to be across the board. We don't get it bad in Arkansas until late in the fall, or late in the summer, and it's across the board. But early on, we'll see it start to pop up a little on things that years where the cold snaps made the plants hold their leaves more. We see it on those leaves that are sticking around, so that inoculum's right where it needs to be.
and that might be every year in South Georgia.
It
But
could
yeah,
be every
we
year.
see it that way. Yeah, early in the season.
The orange rust Navajo is the only thing we've seen.
Yeah.
But some of the Primarchs can get it, but we've never seen it in our
Yeah, you had brought up something in the past year about seeing it on something you hadn't seen it on before. Was that the Primarchs?
North Carolina, people asking me.
Which one?
The Primarch Traveler Freedom, they found a little bit of orange rust.
Oh, yeah, you've seen it on them.
Yeah,
So
it's
they're-
I've never seen an outbreak population.
No.
But Navajo, you find it on this one.
everything I've ever seen for control of orange cane blotch is a 33 frac code.
Yeah.
I have tried like lime sulfur, I've tried copper, they don't seem to do anything to it.
Right.
Is there any concern about disease resistance, since that's all I've ever shown you?
Yeah, so there's always a concern when only one type of fungicide is used for control of something. Now, this is an algae. The group, it's now group, I think, P07, but it was 33, that same group of the phosphonate fungicides. They theoretically have less risk of resistance development, potentially, but we really don't know if that's a concern. I would not assume it's a major concern for that particular pathogen, but I really don't know. That's a good point. Yeah, the coppers are what's recommended for that same species of algae that infects blueberries, and supposedly it has some efficacy for blueberry, but trial work that we've done at UGA has never shown efficacy with copper against orange cane blotch. So, yeah. So it could be, so that pathogen kind of lives beneath the surface most of its time, beneath the surface of those blotches. It may be that it's protected from those applications. It seems to be between April and early July when those sporangiophores actually emerge from the plant. Could targeted sprays at that time be more effective? I'm not sure. It is for prophyte, but I don't know if it would be for coppers or other products. We have not seen efficacy with coppers for that. So.
Yeah, thanks, Jonathan, again, please. Next up, we have Bill Klein. He's from North Carolina. He's out on the side that has more caneberry issues, the ones that we don't see as much in Arkansas. But he's going to talk to us about caneberry leaf diseases. And I know we have some people that are very interested in Cercospora, Pseudocercospora, everything like that. So take it away.
All right, very good. Thank you, Aaron. I appreciate it very much. Appreciate the opportunity to come and speak to you all at this meeting. Work for North Carolina State University, and I'm located at the research station in Castle Hayne, which is a little bump in the road before you get to Wilmington, and then after Wilmington you fall in the Atlantic Ocean. So we're right down at the coast, and so that puts me about 2 hours away from the main campus at NC State. Our research farm is mostly specialty crops, horticultural crops, and the big emphasis is on blueberries. And recently I've gotten more involved with caneberries as caneberries have become a crop of interest in that same area. And so sometimes it's blueberry folks who are getting into caneberries, or folks who are doing caneberries sort of on their own. A lot of it's high tunnel. There's in the coastal plain of North Carolina, there's not a lot of caneberry production, blackberry production under the open sky. It's all mostly under tunnels. So what we'll be seeing is mostly tunnel production. So I'm going to go through, I just sort of lumped all the leaf diseases together, so we're going to look at rust again and things like that, but just very briefly, just to give you a picture, sort of a snapshot of what problems we see in eastern North Carolina. So first I want to just throw this slide in because I had so much fun making it. That's one of my old gray hairs laying on a microscope slide. Yeah, and then what you see all around it is bitter rot spores from muscadine grape. And so just like to share this because this is the scale we're talking about. When we talk about spore production and the size of the propagules of these fungi and the way these things spread by wind or by rain splashing, we're really dealing with microscopic organisms, smaller than the eye can see. And I want to emphasize that because so much of our disease control occurs at an invisible level with the fungicides that you're using on all crops, but in particular on the small fruits. You really have to be applying the fungicides before you see the disease, because all this infection process, these spores germinate like a seed and produce a germ tube. The germ tube penetrates the plant or enters through a stomate or a hydathode, and you get infection and you don't see it. Okay, you don't see it until much later. So if you go to the field and you see spots all over the leaves, that's not something you're going to control this year. That's you're behind the ball already as far as disease control. And so just consider that when you think about a disease control on small fruit crops. The thing that has worked for me best on blueberries is carrying a notebook and writing down what happened in that field in this year and then anticipating it the next year. So if you have a caneberry problem, especially leaf spot diseases, in 1 year, be ready for it the next year. And before you see symptoms, initiate that fungicide program. So anyway, that's my little funny slide with my hair. I can't spare many of them, but I put that one on the slide so you all could see the scale of what we're talking about. So just wanted to mention orange rust again. I know Jonathan covered it in detail, but I thought this was an interesting picture. This is orange rust on a wild blackberry in Bladen County. This was just standing on the edge of a blueberry field, turned around and saw this very picturesque orange rust infection on wild blackberry, just an arm's length from the blueberry field, and took a picture of it. So these are fungal pathogens that are endemic to our area. They're in the wild. And as Jonathan mentioned about the orange rust, it's a real challenge to clean it up around your farm so that you don't have an inoculum source that's close to the blackberries. But I think it's worth doing for sure. Just another shot of orange rust. This is on Navajo, and there was a mention here from someone about Navajo being really susceptible. I would sure agree with that to this rust disease. And this is just going right down that Navajo row, and really hate to have to tell someone that's not going to get better. You know, that you have to take that plant all the way out, and probably the plants on either side of it, because you probably got some movement through. I don't know if it goes through the root system or not, Jonathan, but you'd wonder if it did, if there was any root grafting or anything like that.
I usually spray herbicide in it, just to try to knock everything out.
Okay, so just.
I don't know if I'm supposed to or not, but I do.
Yeah, it's something that you see when you see orange rust in the field. Very often you go back later and you have a hole where you took a plant out, and then two more, on either side. So if either it wasn't dug all the way out, or just be aware this is something to stop for. If you see orange rust, that's not something to say I'll come back and get that later. That's the time to get out of the truck, get off the tractor, flag it, and come back and get rid of it as quickly as you can, because it's not going to get better. It's just going to spread and get worse. And I've been in blackberry fields with folks, and there's a real education mission for us in Extension. I've been in fields with growers who said, "Yeah, I need to spray. I saw that the other day." This is not one you can spray for. So be sure and take it all the way out, and be really vigorous about roguing it. Just another thing we see as a leaf symptom in blackberries in North Carolina. This is the sort of mottling that you would see from a virus infection, and this is another thing that is not going to get better. So this is a plant that has to come out, and very often you'll see this start very small in a planting and then spread throughout. So if you see virus-like symptoms, get it diagnosed. Make sure it's not a herbicide or something that might be a lookalike. But definitely want to go to that planting and get rid of it. I think we see a fair amount of virus material coming in on plant material that was not clean to start with, and so it doesn't take much to wreck a planting, and it's a really expensive thing to try to rectify, where you wind up taking out all the plants under a tunnel system and trying to replant and trying to make sure all the old material is dead so you don't carry over any virus. So just another leaf symptom that you may see. Another leaf disease that Jonathan's already mentioned is the cane and leaf rust that we see in this picture. This is the rust that you can spray for. I rarely see powdery mildew. You see I've got sort of a list there. The two we're going to talk about in a little more depth are Septoria and Pseudocercospora. So we're transitioning now into talking about the fungi that you might control with fungicides. So again, it's something you have to do prior to infection. Once you see a lot of spots, you're not going to get the control that you want. So this is the first one we'll talk about. This is Septoria. I've got a picture of this on blueberry and also on blackberry. The picture on blackberry, the only Septoria I have been able to find has been on wild blackberries that are not sprayed. So that's a wild blackberry. Those 3 or 4 leaves there on your right. That's Septoria symptoms on wild blackberry, and very similar to what you see on blueberry, with that dark reddish ring around the spot and a pale or gray center. And you can often see the fungal fruiting bodies right in the center of that spot. Where it's gray, you'll see little black pepper spots where that fungus is emerging. So this is Septoria. If you compare it to Pseudocercospora, we've still got Septoria showing there on the right, Pseudocercospora on the left, and you see the same sort of pattern, the same sort of size of spots, but the spots do not have that red ring around them, or that dark ring around the spot. They're very diffuse. If you look at the spores of these two fungi, they're so similar, you can mix them up, but if you look at the way they appear on the leaf, the way those fruiting bodies are developed, those are very distinctive. So I'm not sure it makes a huge difference which one you've got. We really emphasize sort of the same fungicide spray program for them both. So I'm not going to deviate there from on the spray program. But I just want to say the Septoria has been really rare for us, and most of what we were calling Septoria was Pseudocercospora. So that's our leaf spot. And Jonathan, I'd love to know whether you see a lot of Septoria or a little or none, or.
Like you said, we almost often don't distinguish between the two, but
Yeah.
we see a lot of Pseudocercospora.
Okay. So this is Pseudocercospora leaf spot. It can be really severe. This is even under tunnel production. Before the tunnels are skirted or covered, the foliage is exposed to rainfall, and this disease can spread, and it seems to be really difficult to control. And when it's severe enough that you start defoliating the plants during harvest, then you can walk out into those rows and taste the fruit, and the fruit just has not developed sugars. It's really not marketable. So it's a really a devastating leaf spot disease when it is severe. The worst outbreak we saw of it was in 2018 when we had just a tremendous record amount of rainfall in Wilmington, and that just really kicked off the Pseudocercospora in that year, and we've seen it since in the wet years. So this is another example of that. It seems to be strongly correlated with cultivar. So some of the cultivars that were developed on the West Coast are really susceptible to Pseudocercospora. I can see that being, maybe because where they were developed, there's not a lot of rainfall, there's not a lot of disease pressure, and so you might have a cultivar that does great there. But when it comes to coastal North Carolina, and you get into our hot, humid climate, a lot of rainfall, then these susceptibilities start to emerge. So when you grow cultivars that were developed for Mediterranean climates, just keep in mind that they haven't met these diseases, but they will if they come to Georgia or North Carolina. And so that's the struggle here, is if you have a susceptible cultivar, it becomes a real struggle to control these leaf diseases on those cultivars. So what we did with Pseudocercospora, and I don't think there's been a lot of fungicide work with this pathogen, or not that I could find, but this is the only trial that I've done, and it was back in 2019. But really as I said, we had a blowout in 2018, and really felt like we needed to have better information about how the fungicides worked. So this was a 5-year-old blackberry planting. It was planted on 3 feet in the row and 9 feet between rows, randomized complete block design with 5 replications. So it's a really robust design for evaluating these fungicides, and I really have a lot of confidence in the results that we got from it. We applied the treatments in the spring prior to harvest, and so that was at 2-week intervals. And so April 16th, April 30th, and May 14th, and that corresponded to our floricane lengths of lateral lengths of 2 to 3 inches, 6 to 12 inches, and pre-bloom. So this was really trying to get ahead of the incidence of the leaf spot. At the time these sprays went on, these leaves looked clean. So we're not doing damage control like late in the season or during harvest. This was trying to get in there really early. So I feel like the timing was really good. We evaluated leaf spot incidence and severity on the 10th of July, and then defoliation on the 8th of August. And when I conduct leaf spot trials, whether it's on blackberry, blueberry, whatever, these are the metrics that I use. So incidence means how many leaves had at least one spot. So severity means once you reach 100% incidence, how many spots were there per leaf. So you were trying to get a measurement of how often infection occurred and how severely it occurred. And then the final evaluation, the defoliation, is when the leaves are diseased and the disease is bad enough, the leaves just fall off. And so that impacts the overall health of the plant, and it really makes it hard for the plant to ripen the fruit. So defoliation becomes a really important metric there. But as you can see here, it was rated after harvest. So and these are the results, and I hope you can see this slide okay from where you are. Older I get, the harder it is to see even here at the podium. So you have these fungicides, the Luna Tranquility, Rally, Orbit, Switch, and Kinja were the ones that we thought might have the best performance, and this is the treatment we used and the amount we used per acre in that first column. The second column is the average number of spots per infected leaflet. So that would be, for a given leaf, how bad was the average number. And so you see the untreated check, we had average around 34 spots on each leaf. So that's pretty peppered up. That's a rough looking leaf there. Best treatment was Orbit, 1.8 average number of spots per infected leaflet. And then the next percent of leaflets with one or more spots was really striking. The untreated check, basically 82% of the leaves had at least one spot on them. Then you go down to our best treatment, which was Orbit, and only 6% of the leaves had any spots. So really felt like this cleaned it up really well. If you go over to the 8th of August and look at the percent of defoliation on these treated lateral shoots, spring laterals, and so this is kind of a careful description because different parts of the plant will defoliate at different times. And so this was an evaluation only of those laterals that were present when we were spraying. So it doesn't do any good to evaluate something that grew out after the sprays stopped. So this is just on the sprayed part of the bush. Defoliation for the untreated check was 94%. So basically those laterals that were present in the spring lost all their leaves. And then you look at the Orbit treatment, we had 13% defoliation, which means they pretty much held their leaves. So this was a huge difference. Felt like this was a big win in terms of which fungicides worked best. So I was really pleased with this trial. But there's a caveat there. There's a little problem with Orbit or with propiconazole, which is you can only use so much of it, and before it starts causing a plant growth regulator effect on the blackberry. So we'll take a look at that. Again, this is Pseudocercospora. The other thing we decided in this trial was that maybe the volume of water that we've used for blueberries, it does not translate well to blackberries. With blueberries, a lot of our fungicide sprays go out before there are any leaves or before there's full leaf cover. And low volumes work pretty well. So you see folks using 25 gallons to the acre or 30 gallons to the acre, or 50 gallons to the acre with fungicides and getting pretty good control. With the blackberries in this system, once that canopy closes in, you just don't have the coverage and don't get the disease control. So the fungicide spray volume, I feel, is really critical for coverage. And I think that's true of muscadine grapes as well. You just have to get a lot of water out there to get through the canopy and cover both sides of the leaves and all the fruit. So really encourage you to use a high volume of fungicide or spray water in the tank. Now this is the treatment effect that we saw with Pseudocercospora. What you have here on on your left is the the untreated control, which was pretty spotty, and those leaves tended to fall off. The one in the center was our result for myclobutanil, and that's a sort of representative of how that fungicide worked. And you see a few spots there. The leaf is pretty much a normal shape and reasonably good control. And then on the far right is propiconazole, and you can see the leaf is a little bit puckered. It's sort of thickened. Propiconazole tends to have a greening effect and a shortening of internodes, so you will tend to get a little bit shorter laterals with it, and the leaves are not deformed, but they're just a little bit different from normal. So the more of that product you use, I think the more it would cause a problem, and you would eventually get to the point where you were stunting the vines. So as far as our recommendations for control, I like propiconazole, but I want it alternating with other fungicides, and I would not use it more than 2 or 3 sprays in a season. And I think alternating in there, it's a powerful fungicide. It does a really good job, and I think it's a useful tool if you have these problems. Now, all that being said, there's a lot of blackberry fields that don't have a lot of leaf spot pressure. So you'll have to be your own judge about how big a threat this is. I always tell folks, don't spray for a problem you don't have. So if you don't have a lot of leaf spot pressure, then focus on the other things that you know you do have. Focus on the botrytis or the other fungicide applications for other diseases, and don't worry as much about the leaf spots. But in situations where Pseudocercospora is a big problem, I think you really have to have an early fungicide spray program, multiple products, and don't let up, at least until harvest. Now, one more slide. I think this is my last one that I threw in there. This is what the canes look like the day I went out to set the trial up, 2 or 3 weeks before we ever started spraying. And what I want you to notice here is how many leaves made it through the winter from the previous season. And so pretty high chance that these are infected with Pseudocercospora, and they're old leaves that came through the winter. And so this is a lot of inoculum, a lot of spores that came through the winter on old foliage that are right there for when the weather warms up and your new shoots come out. And that may be a bridge from last season's disease to next season's disease if you have these old leaves. Now, I don't have a solution for you, but it's food for thought. You know, if we could defoliate this as completely as possible, maybe a winter spray, maybe a lime sulfur. I don't know that lime sulfur would take them off. Whether a product like Dormex could even be used with blackberries. But if you have these a really severe outbreak of Pseudocercospora, I want you to think about how much of it you might be carrying over through the winter and ways to mitigate that. So that's research for the future, but just a thought to leave you with. And that's all I've got. Glad to answer any questions about leaf diseases on blackberry. All right.
Not really time for questions, so you can't run off yet.
No.
I think Wayne had his hand up first.
Okay. Yes, sir.
I know some of my lunar tranquility and rally resulted in similar performance on the Pseudocercospora. If you put those two products together, you expect that to improve?
So the question is, tank mixing rally with Luna Tranquility? Yes. I mean, I think that was, I don't know if it would be synergistic so much as just maybe additive, you know, as far as control. Anytime someone asks me about a tank mix, I know who I'm talking to here, Wayne. You know, tank mixes are a brand new chemical that nobody's used before and nobody knows anything about, right? So if you do a tank mix, don't spray the whole world with it, right? Because you've created something new that might be better or it might be phytotoxic. So products that I have done a lot of mixing with, you know, like we very often will mix these Frac 3 fungicides with a general protectant fungicide like Captan. We do that all the time. I have a lot of confidence in that. But when you start taking multiple modes of action and mixing them together, just be careful. How was that? How was that for hedging? Okay. Yeah. Yeah. Sarah.
Yeah, I've been toying with this idea. What do you think about urea as a way to kind of knock down, like we do this in apples, right? So we'll go out and spray urea either in the late fall, early winter, or in springtime, basically, so that we can decompose the leaf litter. Any concerns about something like that in blackberries?
I don't know. There is debris. There's some leaf litter. The blackberry leaves tend to break down pretty quickly. But you're talking about using urea on the orchard floor
Yeah,
to.
we also spray it in the canopy too. Just to
Oh,
try
really?
to-
Okay,
yeah,
okay.
just kind of knock it back both. Yeah, definitely
Okay.
the orchard floor, but we do some direction up into the canopy.
Okay. Do you spray the whole tree? Just pretty much.
It
Okay.
depends on if we get out, when we get out there, right? We want to hit it on a warmer day.
Yeah.
So the microbes are stimulated by the nitrogen.
Yeah.
Chop it down.
And you don't get it's not like an instant defoliation like a shock thing. It's more just helping it to decay
Yeah, I mean, we've
more
thought,
quickly.
Yeah.
Yeah.
I know that Dalton's tried, Dalton Rhodes has started
Okay.
to do it. You know, I
Yeah.
just don't know that right amount of nitrogen that you can really screw things up, you know,
Right.
especially in the winter, you know, if you do a late fall one.
Yeah. I guess the blackberry leaves that overwinter are so still lively and still so firmly attached. I don't know that the urea would work. You almost need something to shock it off, like a cotton defoliant or something like that. You know.
Good question, you know, like we've seen
yeah.
the leaves just, or the petioles just hang on to the poncus too, and you're like,
Right.
is there a way to get these off, you know?
Yeah.
So that-
And that becomes you also see infection at every leaf.
This is when I start bothering horticulturists.
Okay.
along the way.
Yeah.
I think that would scare me is that I think like in Mexico they force breaking of dormancy with that.
Right,
Same
but that's
type
what
of
I mean,
method.
what is that, and, or is there something else, but what is that fine line, right? I mean, I know in apples, that 40 pounds doesn't do anything, as far as get the tree stimulated for growth again, right? And you ramp it up, but, you know, you have to work out that rate.
I know we want to take a steady, deep, or good, steady drop into dormancy with leaves there, because I know there's been research to show if you get early defoliation, you don't overwinter as easily. So that's what would worry me about trying to knock it all off a little early.
Right, and you know, photosynthesis, yeah, exactly.
Yeah. I think there's work underway as people try to go grow blackberries or certain varieties further south. I think there's probably work underway with products like Dormex, you know, the hydrogen cyanamide and so forth. But that's a product you have to be really careful with, because it'll damage blueberries for sure. I know if you use too much or put it on at the wrong time. Yeah.
You know, while we have it, I have a question in a little bit. Have
Okay.
you seen much orange rust in Vaughn? I would ask Jonathan, but I know Vaughn hates the heat, so I didn't ask him.
Yeah, I don't guess I see enough Vaughn to know. What do you think, Sarah? There's more Vaughn in western North Carolina.
I was telling Aaron that, you know, it sparked my memory that we had, didn't you have an Impala this year, orange rust? Yeah, at the research station, and that one, it didn't get severe. You know, that one surprised me. They're having a hard time hearing me. Come on, guys.
You're gonna move closer.
Come on. But yeah, you know, and then Hannah also told me, she's with Tom Kahn in his program and Gina Fernandez's, and they looked at 10% urea and it burnt the leaves, but they remained stuck on the plants as well. So they've been looking at some defoliant options as well there. Okay, great.
Yeah,
Special Mike, you want to add something, Jonathan?
I was just going to ask, on your trial, I didn't see, you didn't include any QOI fungicides in that trial, did you?
No, there was a reason for that. That's what we were using before,
Yeah.
and well, they weren't working.
So yeah, we confirmed a few years ago that we have QOI fungicide-resistant Pseudocercospora in Georgia
Okay.
at many locations. So I was just going to bring that up. 'cause
That's
that-
really good to bring up. Yeah,
that's-
because that's,
I think
QOIs are such.
5 out of 6 locations we surveyed had QOI-resistant
Yeah, I totally
Pseudocercospora.
should have had that in
So-
here, because that's why we didn't have them in the trial, because they weren't working. Yeah.
You can, just ask. He'll repeat it.
You mentioned that the orange rust happened, you said removing that plant, and if I remove it, and can I, replant it, the new cultivar in that same spot, or you need it to be a certain period of time to clear up the
Question is, if you have the systemic orange rust, and I may let Jonathan run with this, or I'll be glad to answer it. If you have the systemic orange rust, you take a plant out, can you plant back in the same spot with a new cultivar. And I think you can, but that's kind of a tricky question because you really have to get all of the old plant. If there's still root pieces in the ground, you'll get, for instance, if you took out a Navajo plant and you left some roots behind, that's eventually going to sprout, and it's going to have orange rust, the new sprouts from those old roots from that infected plant. And so it's hard to replace a plant. But if you take the whole row out, then you have a better chance of taking out all of the orange rust that might be in the root system or in the old crowns, get all that out of the ground, and then plant a new variety. I think you can go right back in the same spot.
So what you recommend is just in case, if I remove it and leave it that spot, how long?
Well, I think if I was going to plant right back into that spot, I would take out the symptomatic plant and the two on either side of it that didn't have symptoms. So I'd take out, if I had one symptomatic plant, I'd take out 3, and then I'd plant back in that spot. Blackberries grow so fast, there's no reason to leave those 2 that might be infected. So just go ahead and take out a little bit beyond where you know the disease is. You know, it might be 6 plants in a row, right, that all have it. So maybe take out another plant or 2 beyond that. Have a nice clean bed that you have confidence in, and then plant back in that row.
Time for one more question if somebody has it burning. Let's thank Bill.
Okay, very good. Thank you.
Up next, we have Sarah Villani, NC State plant pathologist, on ornamentals in blackberries and apples.
I don't know. I've got ornamental, peaches, blackberries, grape. I've got wine grape. Do I do much wine grape research? No. Ornamentals, yeah, but I've really decided I enjoy well, certainly the apples, but the caneberries is a really nice niche. And I've kept learning more and more, and Bill Klein is a great mentor as well. So that's also been a huge help here. So today, I'm going to give you guys a research update on Fusarium wilt of blackberry. Anyone in here nervous about it? All right, good, because you should be a little bit nervous about it. I've never seen canes go down that quickly from such a disease there. But yeah, I've got a graduate student, Jamie Lanzalato, and so she has been doing quite a bit of work evaluating different strains of Fusarium, as well as different commercially available cultivars, right? And so that, we've done some work, and we know some of the Driscoll's cultivars certainly are quite susceptible to Fusarium wilt, right? But we really don't know what to recommend to the average grower that doesn't grow for Driscoll's, right? And what can we tell you to put in there, at least for now, as we start to evaluate more and more cultivars and kind of work through this. All right, so let's go ahead and get started. So Fusarium wilt is caused on blackberry, is caused by a fungus called Fusarium oxysporum formispecialis mori. That's a lot of stuff to say, so we're just going to call it FOM. So if you hear me say FOM, that's the kind of Fusarium that goes ahead and infects blackberry. There is no evidence that the FOM can go ahead and infect raspberry, at least not that I'm aware of. So this is strictly, as of now, a blackberry problem, okay? And there are other Fusarium oxysporum formispecialis that infect other hosts. And so what is a formispecialis? Just think of it as a specialized strain, okay, that's targeting a host. So I don't know if you've heard about Panama disease in bananas, but that's right over here. It's caused by, Formispecialis cubensi. Okay, certainly we have it in tomatoes, Fusarium issues, and Fusarium in cotton as well right there. And what's really, the symptoms are somewhat similar, and that's because Fusarium can go ahead and clog up the vascular system, the xylem. Okay, so a lot of the symptoms that we're going to see associated with Fusarium are wilt problems. And you might say, well, how do I know it's not Phytophthora or water stress? And I agree. Some of these can look quite similar, the symptoms, right? Just to just to give you some other ideas, we find certainly if you've grown strawberries, you're aware of Fusarium rot, right, or Fusarium rot in strawberries as well, okay, right in the crowns there. But let's go ahead and kind of move forward, and I'll go over some symptoms. Okay, so Fusarium oxysporum was first found as a causal agent of wilting in Blackberry. And that was found in Mexico back in 2011, and I believe it was reported in 2015. Okay, so now, gosh, that's about 14 years ago. No, that's not that long. 10 years, yeah, 14 years ago when it was first found. So yes, it's fairly new, but it's not that new actually. And then in California, it was first reported in 2011. I believe it was identified in 2007. Also as this Fusarium oxysporum causing this wilting of blackberry. And if you know blackberries, Jonathan Oliver did a great survey on different fungi that are causing cankers and decline in blackberries. So there's a number of different agents that can cause a canker and cause wilting type symptoms or flagging type symptoms, right? And so forth. So, but anyway, they found Fusarium as this problem. And then my colleague Bill Klein, he just spoke, right? And gosh, Bill, in 2015, I believe it was, that you all found Fusarium oxysporum was identified as the causal agent of wilting in blackberry. And then, by then, mori was identified as this new strain that's really specific at causing infection on blackberry. Okay, so that's sort of the history there. Back in 2020, as we were all starting to suffer from COVID, I had just actually been appointed a tenure track position here at NC State. So originally, my position was non-tenure track. I didn't have Caneberry responsibilities, but oh, how exciting. I got this phone call from the disease clinic in March of 2020, and you know, they said, oh, this is, you know, we think we found Fusarium oxysporum. They didn't say mori, I don't believe, from the clinic. And that was at an organic planting out in Chatham County, which is in the Chapel Hill area itself. And so I sort of didn't even realize the extent of the problem. But now they found it out east, near where Bill is, in the High Tunnel production. We found it in Chatham County, which is near the main campus of at NC State near Raleigh out there. Okay, so let's go ahead now and just look at some symptoms. You know, these symptoms don't look overly diagnostic, I would say, at first, right? You see certainly decline of the plant, particularly the floricanes, but we also find it on the primicanes. You can see flagging here for sure, and so forth. And if you go in and you can see this dead, you know, the dead vascular tissue as well, because of the pathogen colonizing the vascular tissue. To me, this right here, and I think these pictures were taken by Bill. He's always on the front line of all of this. But this is the most diagnostic symptom for me, is this straight line of black, okay? And we call this this dark brown to black streaking. And no matter where I go, this is, to me, the number one symptom that's like, yes, that's Fusarium wilt of blackberry. I don't see anything else that I'm aware of that causes these type of symptoms. So actually, that place that I had mentioned out near Chatham County, the 2020 place, you know, I wanted an organic field to kind of do some additional research, so I called the extension agent out there. And she says, oh yeah, we've got all the streaking. And we went out there, and gosh, were they Navajo? Oh, maybe Natchez. Maybe it was a Natchez, actually, that it was. But they were absolutely destroyed in that organic planting out there. So we have it at this point out east in North Carolina, as well as in the central part of the state, okay? And then the other symptom that could be quite signs and symptoms that could kind of help point you in the right direction here are these great pink to graded pink sporulation on the floricanes. So there's some examples there, right at where the petiole, the junction is on that side, and so forth. What I will say is that Calototricum species can also infect blackberry, and so I think these spores sometimes look quite a bit similar to an untrained eye out there, or even to a trained eye. I'm getting better at it. But just kind of keep that in mind as well, that, you know, you need to look at the whole picture and then say, oh no, we might have a problem. Let's go ahead and contact your disease clinic or the specialist or your extension agent and get that in for us to do more of a workup, okay? All right, and that's just, I think we saw some of these photos earlier where you have complete collapse of the vine. So funny story, I don't know if it's too funny to the growers, but I was called out, out in, was it Cleveland County? So in the foothills, right, about a couple hours out near Shelby area of North Carolina. And so there were 2 growers, and one of them, I think it was Primark 45 that was ordered, I want to say. But anyway, one of those growers had planted, and within a month or 2, all of the plants had declined from Fusarium wilt. And we had another grower down the road that kind of split that order, and they ended up turning out okay. And so, the question is where is this Fusarium coming from? Is really one of the main questions. It has, if we start to sequence this FOM, it has a very similar sequence to the Fusarium that infects strawberries, So could there have been some, change in the gene that now we had a jump from strawberries to blackberries? Maybe. It's kind of like the Neopistolochiopsis issue. Where did it come from? Was it shipped in on other plants and established? Maybe. But what I will say is that what we found out in the Cleveland County area had similarity to the original Mexican isolates of FOM, and those out east had similarity to the California isolates. And so there looks to have been at least 2 different of introduction events with that. Okay. So why should you even care about this? Really, the main issue is we don't have a way to control it. It's not like I can go ask Brianne over at BASF to give me a fungicide, because nothing has worked so far that we are aware of to really go after this. We can start to borrow from other production systems that maybe some SDHIs might work, but nothing is labeled, nothing is registered, and it really would be a reach. Okay? At the same time, fumigation hasn't worked all that well. They're using it on strawberry, but why not on blackberry? I think there's 2 different reasons going on there. First of all, I think you really need to get that fumigant deep down. You've got some pretty long blackberry roots compared to strawberry, I would say. The other argument is that, these blackberries are going to stay in the ground for more than 1 year. And so even if you go ahead and fumigate, I've spoken to Bill, and we really do believe that those spores that we see are going to be aerially dispersed. So they're not just traveling through the ground, they're kind of traveling through the ground, up above and everywhere. And so, a fumigant itself probably just isn't going to hang on and do the trick. So, we're kind of out of options with that. But, our blackberry industry is increasing. The last figure I looked at was, valued at $12 million in North Carolina, but that was 4 or 5 years ago. It's certainly more now, and I'm seeing other states go ahead, put more blackberries in, so yeah. I said that already. And then the thing about wild blackberries, okay, Rubus alleganensis. I have Mayact as a host reservoir. You're going to find out in some of the research that we did that it is a host reservoir. So basically, what the heck does that mean? What it really means is that I can have wild blackberry, and it can harbor asymptomatically the FOM pathogen. So that's a problem, because I don't know, I'm going to talk about our survey, but everywhere we went, there were wild blackberries everywhere, right? And so it's really impossible to go ahead, I would argue, and eradicate all of those and get rid of them. And so that's another deal that we need to worry about. And they found that wild blackberry out in California was also, their species of wild blackberry was also serving as a reservoir host. Meaning, again, it can hang out asymptomatically, the pathogen, but still cause problems. All right, so kind of keep that in mind. Okay, and then several cultivars are highly susceptible to Fusarium wilt. All right, and when we started this project, we knew what cultivars tended to do bad in the field just because we had gone out east with Bill. And Bill had told us, and we had observed this. But at the same time, a lot of the growers out in eastern North Carolina were collaborating with Driscoll's, and so they had proprietary cultivars as well. And so we really didn't know the scope of, what commercially available cultivars, would be susceptible, or if they all would be. All right. So now I'm going to go ahead and transition to some of the research that we've been doing, and some of it gets a little bit heavy, but I'll go through it, and we'll see how that goes. The first goal was really to conduct an expansive sampling of cultivated and wild blackberries across the state. Done, and I'll talk about that. Then we wanted to conduct a phylogenetic analysis of these isolates. All right, why would we want to do that? Okay, so here's the deal, all right? We want to see what the dominant strains of this Fusarium oxysporum mori, this FOM, are available. Why? Are all of those genotypes that we look at causing a problem? Are all of them pathogenic? Are some of them pathogenic? You know, are all of them highly aggressive, or just some of them highly aggressive? Are we seeing some strains infect some cultivars and other strains not infect other cultivars? So that's really where we wanted to get at, is just to start to create a map of what that looks like, and then hopefully we can inform our breeders, you know, hey, let's take a look at this strain on these cultivars and get a better idea of how to go about this, okay? So then, yeah, we conducted pathogenicity and aggressiveness assays on select isolates using a highly susceptible cultivar, and we chose Primark 45, which unfortunately, you know, has the thorns on them. So that was really cruddy to do, but it tended to be the most susceptible cultivar that we were working with. And then after that, we wanted to take select isolates with different levels of aggressiveness and then test them on 6 commercially available cultivars, you know, yeah, grown in the southeastern United States there. All right, so that was the plan there. Okay, so how did it start to work? Well, Bill hosted Jamie and others from my lab for a few days and took them around eastern North Carolina. And we looked at 3 plants from each infected cultivar sampled per farm. All right, and just observationally wise, Primark 45 and Monica seem to have more Fusarium wilt than Wachita, and the severity on Wachita seemed to be greater than Victoria, Natchez, and Galaxy. Those were just quite observational type things on that. And so forth there. And then we looked at the roots, the primicanes, and the floricanes in June and July. And even if they were asymptomatic parts of those, we still went ahead and sampled them because we wanted to see where the pathogen was in the plant. And then from each of those farms, we looked at wild blackberry, Rubus gallaghenensis. I had my colleague, or actually she was my technician at the time, Pierce Lynch. She's now doing a PhD. But she took her machete out there and came back with poison ivy and chopped through the bush. They took me to do it one time, and I ended up with ticks all over me, and told Pierce I would give her a raise after that, because it was pretty brutal with that. But she was prepared, and she could identify, you know, quite easily the different weeds out there. I mean, I sometimes couldn't tell you what poison ivy looks like, to be honest, so that was really helpful. Okay, so we collected samples from 9 farms across 5 counties in the state. And then those wild blackberries from each of the farm. And then what we did is we looked at 2 different regions within the genome, 2 different loci they're called, that had been done in California, so we already had reference sequences to which to compare those 2. All right, so these were the positive hits, as you can see here, Pender County, Sampson and Chatham, so kind of the central to the east, and then here in the foothills as well in the Lincoln-Cleveland County area. Okay, so those were where we had positive hits. All right. Now to show you the work-through of how this all goes on, basically we get the primicane, the roots, the floricanes, and we go ahead and we do something called surface sterilization. Why do we do that? Well, we do that so we only get what's inside the plant and don't get any of the other crud that's on like the root outside of the plant. Okay, from there, you go ahead and you plate that on agar, and then you get all of these different types of cultures. And not all of them are Fusarium or look like Fusarium. There's endophytes, there's Botrispharia, there's Phomopsis or Diaporthe. There's a whole bunch of stuff in there. But our goal was to really go ahead and zone in on anything that looked like Fusarium oxysporum mori. Okay, so we got those, and then we extracted the DNA. We targeted 2 regions in the genome for DNA sequencing, and then we sent it off for sequencing and compared those sequences of our isolates to what was going on in other places. Boy, that gets complicated, but I just wanted to show that to you. So that's the gene, one of the genes that we looked at there, okay? And where you see these little blips here, that just means it's different, right? And so we're working out to see how different is it, look at different strains and so forth. But we found 9 different genotypes within that region that we looked at, and we got, from symptomatic, Primark 45, Wachita, Natchez, Victoria, and Monica. Okay, so we found FOM on all those cultivars. We also found FOM on Galaxy, but that was asymptomatic at the time. And then also wild blackberry, we were able to isolate FOM from as well. Okay, cool. So then we went ahead and did some research here, basically. And the first thing we looked at was we wanted to test those different genotypes. Are they pathogenic? Do they cause disease or symptoms in a highly susceptible blackberry cultivar? Again, we use Primark 45. And are there differences in aggressiveness? Okay, so that's two different things. The first is yes, no. Do you cause disease, do you not? And the second question is quantitative. How much disease do you go ahead and cause? All right, so let's go ahead and we'll show you how we did that. Okay, Phylotech provided us all of the plants. And so we washed off the plants, got them out of the soil, washed them off. We went ahead and cut the roots. Yes, that was stressful, but we wanted to standardize the roots before we went ahead and went forward. Okay? We then went ahead and made a spore solution of each isolate, put it in this high-tech shaker here with mason jars, very, very classy here at the Mountain Horticultural Crops Research Station out near Asheville, and then we put them in the greenhouse. Okay? And again, single cultivar, 9 different isolates. Actually, 14 different isolates, 9 different genotypes. We then went ahead and gave them all a rating, 1 through 5, and then I had to back transform it. So basically, he got a rating of 1, it was asymptomatic. A rating of 2, you can start to see some reddening and yellowing or chlorosis. Okay, that's about 1 to 33% disease incidence, I'd say. A 3, more reddening. You start to see dying lower and upper leaves there. 4, still hanging on to life. And then 5 was giving up the ghost at that point. All right, so that's how we went ahead and we took 8 different evaluation dates. The dates here, we tried to do it weekly, but then there was a hurricane and we couldn't go to work. And so for a month, although Jamie did sneak in, we weren't supposed to go to work. So we had 2 weeks of not taking any data there. We also looked at plant height, but I'm not going to show that data today. Then we were curious, you know, I'm very fortunate to get to work alongside a horticulturist, apple physiologist, who also does work on caneberries at the research station. So NC State went ahead and spent hundreds of millions of dollars investing in a new plant science initiative and a new plant science building. But quite honestly, you know, the goal with it was to bring people from different disciplines together. But like I said, really, I'm very fortunate that I've got somebody that I work with regularly and that we can do this kind of collaborative, cross-disciplinary work. And so that's what we did. We used the spectrometer to go ahead and evaluate spectral data. I won't be showing that today because of time, but the hope was, can we sense a stress plant from Fusarium, or a stress plant in general, before symptoms start to show up, right? That was our goal with this. Why is that important? Well, maybe we can eventually go ahead and use sensors in the fields or drones and kind of start to detect bad areas and so forth. And with some of the type of the spectral data, it can tell, I guess, a disease versus water stress and so forth. We haven't, right? Is that right, Hannah? It can kind of go ahead and tell, right? There's different wavelengths, say this is caused by disease or this is caused by this abiotic stress, right? And so we haven't analyzed this yet, but we're looking forward to that. And then we also used a LICOR machine to measure something called stomatal conductance. So the stomata, as you can see here, this was provided by Melissa Munoz, Tom Kahn's postdoc. But what you can see here is these little structures here and here. Those are stomata, and they open and close. In the simplest terms, it helps with water exchange. It's a lot more complicated than that, right? Water vapor exchange, but it's a lot more complicated than that. What we should see is that the more aggressive the isolate or susceptible the cultivar, the lower stomatal conductance. Okay? And then we also looked at something called photosynthetic activity. And really in layman's terms, that means how efficient is the plant converting light energy to chemical energy, basically, in order to conduct photosynthesis. Okay, so we looked at 2 of those. So again, the same trend. As the plant, if the isolate's more aggressive, or the cultivar more susceptible, we would expect lower photosynthetic activity. Okay, so pretty straightforward. So let's go ahead, and I'm just going to kind of not go through this too crazy. I don't want to make this too complicated. But there are some take-home messages. So again, this here is the disease severity over time. So we took disease severity ratings over those 8 different dates, And then there's a way, it's called area under the disease progress curve, but there's a way that looks at the disease over time and kind of lumps it into one number for us to evaluate, okay? So basically here, the take-home message with this, is that there are different isolates that have different levels that cause different levels of disease severity on Primark 45, okay? So they're not all similar, they're not all the same. Okay, so here, this isolate here and this isolate here were among the most aggressive isolates. They were also from the two most common strains that we found in North Carolina. Okay, so that kind of makes sense. This isolate here in the light blue, that actually came from our wild blackberry. That's a problem, because the wild blackberry is able to harbor pretty nasty isolates as well, okay? And so what does this kind of look like over time? I took two different isolates, one highly aggressive, one moderately aggressive, and then the control, so non-inoculated is here. And you can see how the disease progressed over time and all of those. So eventually, that moderately aggressive isolate was going ahead and catching up to the severely aggressive isolate. Okay, so it may not matter too much in the thick of things. Okay. Now let's go ahead quickly and look at the stomatal conductance. And again, I just want to pay attention to this isolate and this isolate here, which caused the greatest disease incidence. We also saw some of the lowest stomatal conductance. All right, the statistics didn't pan out for everything, but numerically, that's what we saw. Can we start to use this as a predictor for the disease severity? Yeah, I think we can. So when we look at some of the earlier values, this is stomatic conductance and disease severity, it's really jumbled up there. But right before disease starts to get severe, there's a nice correlation, right, between disease severity and stomatal conductance. So, we're going to look into this more, but there's definitely a relationship there where we might be able to early predict Fusarium in the plant, and so we're going to keep looking at that. Okay, same deal here. Let's look at pathogenicity or photosynthetic activity. Really the same trends as we saw with stomatal conductance. And again, we see that nice trend here, but we were able to catch it even at a lower disease severity, how those start to trend nicely. What does that mean? Again, maybe we can use some of these markers to say, hey, you've got disease, but right before we start to see disease symptoms. So we'll see how that goes. Okay. Next, we wanted to go ahead, like I said, and look at commercially available cultivars. So we looked at Primark 45, Wachita, Ponca, Galaxy, Twilight, and Vaughn. All right? And then we looked at 3 different isolates with varying aggressiveness from our Primark 45 trials, okay, plus the non-inoculated. All right, so, oh man, I still put number of days. I forgot to change that. They were inoculated on election day. So you can go ahead and do the back math, but it's about 2 months or so from when those were inoculated. Jamie let me know last night, and I forgot to change it, but that's okay. So you can see here, this is the moderately aggressive isolate, almost 2 months after we inoculated. Okay, so we've got Galaxy, Twilight, those are both from the Oregon breeding program. We've got Primark, Ponca, and Wachita from Arkansas, and you've got Vaughn here. So it's quite clear already how these are going to start to separate out as far as susceptibility goes. Okay. Proof is in the pudding. Again, this is just looking at one snapshot. We're still doing this trial, so I don't have the final data yet. But I can tell you that, Galaxy, across the 2 really aggressive ice, highly aggressive and moderately, Galaxy, we really didn't see all that much disease. It was below 5% in those cases. And Ponca and Vaughn really started to trend. Now, I will say that these are 2 strains, right? There are other strains out there. That's number 1. Number 2, this was done in a greenhouse. And so what we're going to do is I spoke to Bill. We've got some grant funding from the Specialty Crops Block Grant Program in North Carolina. I mean, I'm happy to go ahead, if Bill's game, and we're going to go ahead and put these out in Fusarium-infested fields out east. I can't put them in western North Carolina because we haven't found FOM yet in western North Carolina. And so I'd like to put them out there, and if Aaron wants to play ball, we can do some similar trial work out there at some point as well. And certainly with other cultivars. They're very easy to screen in the greenhouse. It's about a 2-month process, and then we can watch the destruction happen in the field afterward. But it kind of gives us a nice early screening process. As you can see with the weakly aggressive isolate here, disease severity, really the ponca in that case was a little bit greater, but there were no significant differences when it came to that. Okay, we did again this stomatal conductance, and I don't want to belabor this because I'm actually kind of on time, but you know, we started to see here with the weakly aggressive isolate, there were differences with that. This is relative, so we had a non-inoculated plant. We looked at each isolate compared to the non-inoculated plant. But you can see here that, you know, the Primark 45 in this case actually was one of the better. So, you know, there might be some kind of strain thing. We only sequenced 2 genes. It only tells part of the story. So, you know, it'd be fun to do some deeper genomic sequencing that I don't do, but I know some colleagues that would do that. And so we can do some of that work as well. And the same deal with photosynthetic activity, similar trends. All right, so what are some basic best management practices for now? Look, we really need more field data, I would say. I can go ahead, publish on the greenhouse stuff, and start to get an idea. But there's so many factors, right? These are ideal conditions in the greenhouse. They're not under an insane amount of stress other than the root cutting. You know, they're at a perfect temperature for infection to occur, and it really had to be it really was kind of a nice a very narrow window, actually. So it liked hot. You know, if I were going to plant in an area of the farm, I consider Galaxy, Vaughn. I mean, Ponca did well, but then Bill said, it wasn't that, you know, they kind of collapsed out there. So it'd be interesting to go ahead, let's go ahead and try this in a couple different areas. And then if they decline, we can get those strains and collect them from the plants again and see what's actually attacking each of those cultivars. So I think that would be my next, but I feel pretty good with Galaxy. It looked good when Jamie went out to collect. It looked good in the greenhouse trials, so yeah. You know, not all the strains were identified through sequence data, or not all strains that were identified through sequence data as FOM were pathogenic. So we're working on building that phylogenetic analysis right now. Let's see where everything kind of comes out, right? And I know Arkansas has been sending us too, so let's see how similar those are and see what's going on there. Okay, you saw that different isolates in FOM had varying levels of aggressiveness. So all of those were taken from symptomatic plants, but again, you know, these are ideal conditions. I know that plant stress has got to be a huge factor, especially dry conditions. At least in other symptoms, dry conditions is a vascular pathogen. You clog up the xylem, that's a problem getting water through. It's dry, well, then you don't have much water to get through anyway, and it's kind of a double whammy with that. Okay. Sanitize and protect your feet. Okay, it hangs out on the ground. So if I'm wearing my Keens out there, my work boots, or my flip-flops sometimes, I don't want to go through a field that we know has FOM in it and then go over to another field. Kind of work in your problem areas, right? And go through that. Use clean planting stock. I haven't seen an issue with planting stock yet. Case in point, what happened in the foothills. It wasn't a planting stock issue. They went to 2 different growers from the same source. One ended up with it, one didn't. So as of now, we don't believe it's coming in on any planting stock. No problems with that yet. Of course, remove those diseased plants and destroy the residue, but those spores can hang out in the ground, just like Neopistolochiopsis for those that were in the strawberry talk earlier today. It can hang out in there. And again, we can fumigate, but that's really not all that helpful, considering the perennial plant. I'm really curious. There was a talk earlier today about the possibility of annual cropping blackberries. I think that gives it another level of, if you're during a high density system, what kind of stressors are we going to add to that? It's curious, But yeah. Obviously, avoid replanting in those known infested areas if possible. It's not always possible. And so, what are we going to do? And we're still kind of looking into that, Certainly a different crop in there could disrupt the cycle. It is FOM. It's detecting blackberry, so could a cover crop help break the cycle? It depends on how long those FOM spores are hanging out in the ground also. Okay, it's soil and plant-borne, we believe, so avoid transferring that infested soil. Be careful on shared equipment. Okay, you can pick it up on the tires, move it over to another farm. We've got a new problem here. So be careful with that. And then like I said earlier, work in those clean fields first, or ones you believe are clean. And then move to the uglier ones. And with that, I know Jonathan mentioned the small fruits, all of the chemical guides on there. Bill Klein wrote a great fact sheet of Fusarium wild of Blackberry that's available if they haven't yelled at us yet that it hasn't been updated. And of course our Ag Chem manual, and of course the My IPM app. That's just the day before the technical hurricane, but we couldn't get out to the orchard at that point. A few words of thanks, certainly Gina Fernandez for helping us decide on some cultivars. We would have liked to have tested more. All of the light core, that photosynthesis and stomata conductance data has to be collected in the morning. And so that really limited how many we could test. From here on out, now that Jamie's finishing up, we'll probably just go ahead. I feel pretty comfortable with our rating system and just to do that work. Certainly Hannah Lepsch and Tom Kahn. Tom for providing the light core and the spectral machine, the machine, the instrument to read the spectral data, and then Hannah, because I have no idea how to even begin growing blackberries and what to do. So she's been a huge resource. And then, of course, our temporary techs, Caitlin, who is Jeff Chandler. He was the director of our research station. She was a technician in our lab over the summers, and Pierce, the machete queen. And with that, I think I've got time. I'm over time, darn it, for maybe a question or two.
Yeah, it's
Okay.
great, so let's all thanks.
All right, cool.
We have a 30-minute break, but if you have a burning question, for sure ask it, because, I mean, everybody will have the freedom to move if they want.
Cool. Great, everyone wants break, so, yeah, we got one? Yeah, go ahead. You think you should worry about a mixed population of fawns? Yeah, it's a good question. I haven't seen, definitive enough. This is, we're repeating this trial. Not yet, do I think. I mean, I'd like to see how the phylogenetic analysis plays out, and then see what happens in the field there. I don't know. I don't know if there's specific strains targeting different cultivars. I can't say that yet. Mm-hmm. Yeah. So, great. I'm not going to play that game guessing. It's here now, right? Where do I think it came from? I think you track, you do some barcoding there, and you track maybe where those strains kind of were identical to other areas. That's what I'll say. They just didn't, thank you. There you go. It doesn't help explain the foothills issue where we found the Mexican strain, the strain from Mexico, I should say, but I just don't know. But yes.
We had one pop up on a grower who does not interact with any other growers. 1 acre, just got plants, and he has Fusarium. So I don't know how he got it from California, I can tell you that.
Some of these strains are just so darn close to Fusarium, the Fusarium and strawberry. I still wonder what's going on with that, too. I mean, we may not be sequencing enough genes or loci yet to truly identify that. John?
How often did you find other Fusariums that were not?
We found them often enough, but we didn't. So I have this way of holding on to everything, everything that I isolate from wood tissue, and it gets insane. So to streamline things, I said, let's only deal with the Morai right now. And what looked like Morai and then confirmed by it, but yeah. There were others, and I'm sure they're causing some level of plant decline, but
I just wondered if you found the strawberry.
I'd have to go back and look and see if one of them was identical. Cool. Yeah.
So from my understanding, once you get it into the field, it just keeps spreading. Is that what happens?
Bill, you want to take that as far as you've seen? I've seen, I mean, it's everywhere when I go somewhere.
Once it's in a field, it's hard to get rid of fumigation, and even if you're fumigating, you can only fumigate so deeply. Eventually, the next blackberry crop is the roots are going to get far enough down below the fumigated area, and then they're going to run into the pathogen again. If it was an annual crop system, you could get by with fumigation and grow the crop, harvest the crop, be out in the field. But with a perennial crop like blackberries, you get a benefit for a year or two, and you need a benefit for 8 years, 10 years. And it just goes away, and they get disease all over.
But, I mean, originally we were hoping to pick point sources and kind of spread out across rows and down rows and get asymptomatic plants from that. And when Jamie got out there with Bill to go do the sampling, it was the entire tunnel that was already in collapse. The The Wachita really surprised me, though, in the greenhouse trials, because it seemed to at least move slower, I would say, in the field. But by 2 months, it was just as terrible as almost as terrible, I would say, as something like Primark, 45. So, great. Not that we're aware of. I haven't seen it. I think did California look at that maybe at one point? Somebody looked at some. I feel like I've read it somewhere, not that we're aware of. We can try to put some in the ground and see what happens, but not yet. Cool. All right. Well, thank you very much, everyone. Have a good break.
Okay, so we'll get started. I don't know how long this will take, and so we'll hope to not go over time, but I'm moderating, so I guess we can do whatever we want, right? We won't tell Amanda in the back. So my name's Aaron Cato. I do all of insect and disease management for the food, specialty crops in Arkansas, so vegetables, fruits, things like that. And so I work a lot with insect pests. That's what my background is primarily in, and then I've been drugged a little bit kicking and screaming into diseases by our growers who, have disease issues. And so I'm an extension specialist, so if there's issues, I got to work on it. And so that's why I'm working in nematodes. And so if you can see that picture there, this is a very common occurrence in blackberries. And I think the thing says blackberries, but we're going to talk caneberries, a bit of raspberry, a bit of blackberry, because a lot of the nematode work has been done in raspberries in the Pacific Northwest. And so we're going to lean on that a bit, and then I'm going to show you some data that we have from one trial that we did this year with post-planting applications of nematicides that work in other crops to see how they work in blackberry. So what are nematodes? I wanted to put microscopic, and then I was thinking, I've seen these things come out of grasshoppers all over. They're often microscopic and transparent roundworms. They feed on a large range of plant and animal hosts. They're a pathogen, right, if depending on your perspective. And then most of the species are going to be found in the soil and can often just be very important for soil health. And so whenever someone talks about nematodes in reference to crop, they're probably talking about plant parasitic nematodes. And so these are ones that are going to damage your plants in some way. So whether it's directly where they're actually feeding on the roots and the cells within those roots and allowing entry to things, or weakening the plant, stealing its ability to pull nutrients and water out of the soil, or indirectly where the wounds that they occur or they create are going to allow infection, or potentially they're going to vector or be a host of a virus in some way and then allow that entry into the plants. And that's a lot of what we're seeing in caneberries, where we get a lot of nematode species that are very likely to transmit viruses. And then we also have entomopathogenic nematodes. So these are ones that actually kill insects. And so those are ones that are very important for soil health and are being marketed as alternatives to conventional insecticides. And in some cases do a pretty good job. In others, it's yet to be seen what the value is. A lot of times cost can be very prohibitive. There's a lot of nematodes that impact caneberries. You can find a large list of them. So dagger nematodes, root lesion nematodes, root knot, lance and cyst. I'll tell you that, looking through a lot of the literature and what we've seen in Arkansas with our experiences with nematodes is primarily we're thinking of dagger and root lesion. And I think that would be the same thing they would tell you in like the Pacific Northwest or something, where these are their problem issues. And so what we see a lot of times are virus issues in caneberries and especially in blackberry. And so it's kind of this idea of how quickly are you going to get these viruses in this planting that you hope to survive for 8 to 10 years and give you good production. And nematodes are contributing to the spread, or to some degree, the amount of risk that you have of viruses building up in the plants. But they also directly damage roots of the plant itself, and so that's primarily going to be from root lesion nematodes. Daggers, they'll feed on them definitely, but they're just not doing as much injury directly. But dagger nematodes can transmit tobacco ring spot virus, tomato ring spot virus, grapevine fan leaf virus, peach rosette virus. And what we see is that these things kind of stack up and it turns into what we call yellow vein disease. So we'll talk a little bit about the biology of these two types of nematodes, because I think it's really important in understanding how you could strategize creating an integrated approach to managing them. And I think managing is a key word with nematodes, because I think if you talk to any nematologist, eradication is a word they don't ever want to hear, because it's very hard to get rid of nematodes. But you can work in a direction to where they will eventually potentially go away. It's kind of the idea. Dagger nematodes are external root parasites. This is important because they are not living inside of your tissue of your roots. You know, you hear about root knots, nematodes. A lot of times you have to look inside the roots to find those things. They can survive several years in the soil. That's another thing that kind of sets them apart from other nematodes. They're very long-lived, the adults are, and so they don't really multiply really quickly. They don't build up in numbers very fast, but they are going to stick around for a long time. Viruses are acquired by dagger nematodes throughout their either juvenile or adult stages through what they feed on. And so they don't pass that to their eggs. And so if you have a dagger nematode that feeds on an infected plant with a virus that they can move, they could pick it up. It's not always 100%. But if there is no host of that of the viruses that they can transmit around in their feeding, they're not just going to get those things. And there's actually been some work to show that whenever dagger nematodes feed on grasses, especially adults that are long-lived, they can actually lose some of those viruses over time. When they molt as a juvenile, they actually will shed those viruses through the molts as well. There's some work that's been done to show that. These kind of things, you know, it's not as linear as you expect with some pests. You know, X number of dagger or nematodes feed on X amount of roots and you get less yield or whatever. It's more about a risk, and as the numbers increase that you're finding in the soil, the more risk you have of virus transmission occurring onto Blackberry especially. So these nematodes are just, you know, feeding on the roots themselves, and there's a little bit of damage occurs, but the big thing is they can survive deep in the soil as well. And so if you get rid of all the crops, all the plants in that in your area, right? You rip out the blackberries, you spray herbicide, they will actually move down in the soil to find older roots to feed on. And so there's some issues with them actually moving down, either to feed on blackberry roots that have gone kind of further, kind of like Bill was talking about with Fusarium being controlled easily at the top in strawberries, but not as easily in blackberries because the roots are further down. And it can have some issues with these dagger nematodes as well. So when are the numbers the highest? I'll tell you from our experience in Arkansas, they kind of build through the year. In the Pacific Northwest, they would tell you it's highest in the fall, in the winter, and the spring, the early portions. But the reality is it's a lot of really connected to when we see the most fibrous roots on our plants and when there's moisture. And so if you look at any data from the West, they're going to tell you there's no moisture, right, in the summer. Well, we're not the same. And so we see a lot of these things in the summer because we get a lot of rain. And so I don't think it's as centered around just the cooler months as it is other places. Root lesion nematodes, these are actually migratory endoparasites. So that means they're going to be living inside of the roots themselves. They lay eggs that they're going to put out into the soil, but they're actually living inside of the roots themselves. They have several generations per year. They can move through plant material. So if you get a cutting of a blackberry from a friend, you may be getting some root lesion nematodes from them as well. They can move through the soil, so movement of soil. And I didn't mention that about dagger nematodes, but they can as well. If you are moving soil around or tilling your field, you can move around dagger nematodes, or through the irrigation water, actually, which I think is a little bit less common. Their populations are generally low through the winter, and then they kind of increase as the plants are increasing and pushing out more growth. And they can survive actually in dead caneberry root material for 8 months. There's some research showing that you go in there, spray, kill raspberries, 8 months later they have these bits of roots they were tracking, and they still had living root lesion nematodes on those. And so it can be pretty hard to get rid of. So viruses in caneberry, you know, traditionally we talked about this yellow vein disease, a stacking of multiple viruses, and so it's not that one virus that you're seeing. It could be tobacco or tomato ring spot, but accumulation of 2 to 3 or more viruses, you start to see the symptomology, and you know they're constantly under attack from several different sources that are vectoring the viruses to the plants. And I think there's some good work in Clemson going into trying to characterize what these sources are, but it's largely unknown. We know it could be from areophyte mice, is what we've learned recently, from pollen from wild blackberry, coming in on plants if you're not getting good tissue cultured plants, maybe spread by leafhoppers or whiteflies or aphids. We're not really that sure. But we do know is that they will build up in viruses, so the pressure is high. And dagger nematodes are contributing to that to some degree. And so if you have a whole large population in the field, that's going to kind of amplify the effect that's occurring already. You know, spread of the viruses from nematodes themselves, it's not generally because of a movement of the nematode. And so what they found, especially in the Pacific Northwest where they've done a lot of this research, is that they can track the movement about at the same speed you would expect the roots of the plants themselves to move. So if you're left alone, just nematodes, you could expect maybe 1 meter of movement per year. But whenever you have root movement to other rows, then those plants are infected and they're already over to the other rows. That's when you get these large levels of spread in the field. So it's not really occurring from the nematodes themselves. Now, if you are moving a lot of soil around, which doesn't occur during an act of planting too much, then you can move them. But generally, once you have a set in stone planting, you're not moving the soil much after that. Okay, that's all of that. So how to know if you have a nematode problem? The best time to find out is before you plant. And so most of the strategies we're going to talk about are employed prior to establishment. And once you get them there, there's not going to be very many options. And that's why we're looking at research on it. So the other thing is that high levels of lesion nematodes can lead to poor establishment. And so this is some work they did on raspberry with root lesion nematodes. And when they were getting to these huge numbers in the soil, you're hoping for a lot of really good fibrous root growth to support a healthy plant to survive the heat of the summer, especially in the southeast. You're not going to get that if they're being fed on by a large number of nematodes that were in the soil when you planted. So, you really need your soil sample before you're going out to establish things. So the time of year that's best in Dagger, there's this idea between the fall and the spring. We have looked in the fall, we've looked in the spring and in the summer, and we've seen pretty stagnant amounts. It could be a little different with lower numbers, but we've had success at all times of the year in Arkansas at least. And so I think the fall is generally a good time, especially when you start to get moist soil, but I wouldn't do it whenever you're in the middle of a drought. Root lesion nematodes, they seem to be in the warmer months, so late summer, early fall would probably be a good time to do that. The depth really depends on the nematode you're looking at. For daggers, a lot of times they will be around 8 to 12 inches in the soil. They actually are a little bit further down than some of our typical nematodes. So, somewhere between 6 to 12 inches, I think you're gonna be able to find those things. So what do the numbers mean? You're going to get back different types of tests from different labs, and ours comes back as a cubic centimeter of soil. And so sometimes you'll have to kind of look at the numbers depending on the actual value that you get back. So I would really recommend you work with your local specialist, because I've gotten a lot of different ideas on what is important, what number means what. And that's, we don't have a good definitive answer is what I would say for blackberries. How do you sample for nematodes? You really want to try to capture what is a highly aggregated issue. So you may have a spot here or there that could spread eventually through the plant spreading viruses, but you're not going to capture that if you're going down a row and down another one and that's all you look at. So you want to try to get some zigzag approaches. If you have an area where you know they're at, try to make a zigzag in and out of that area where you're having some of the symptoms. That's usually the best way to go about looking for what nematodes are in a field. I left the wrong title on this slide if you haven't noticed. But so the idea is what number of nematodes are an issue. And so there's no straightforward threshold for daggers, and there's not one that I've seen in blackberries. I've asked several nematologists, and I get several different answers. What I've heard is that in Georgia, if Phil's in here, he can correct me, but in some of the areas around Georgia, if they find 10 or more dagger nematodes per 100 cc's of soil or cubic centimeters of soil, then they're going to fumigate that soil before they put the plants out. So that's 10. I would say in Arkansas we get a lot of that. So probably a lot of things that need to be considered. I've heard from some other nematologists that if you're seeing 120, that's going to be a high risk of virus transmission to your plants out there. And so just some numbers to kind of think of. Root lesion nematodes, there's a lot of established thresholds for red raspberry that I was able to find. And so population greater than 100 nematodes per 100 cc's can hurt establishment. 200 to 800 are kind of an action threshold for where you need to start thinking about adding in some post-planting nematicides, and because they know that once you hit 1,000, they have a lot of data that shows that they have direct effects on the plant health themselves. So what are the control strategies that are often used or often recommended? And I think most people's first thought goes to fumigation, right? And there's different ways that people go about it. There's whether or not you're going to use a tart fumigant or you're going to use row fumigation, right? You can probably see there's going to be a large difference in efficacy when you consider a tart versus a row, right? You have this large area that you just tilled all your soil and old roots through that's not going to have any control measures. So we know that lesion nematodes are going to stick around for 8 months at least. We know that dagger nematodes can live several years. If you're only fumigating one spot, you can imagine you're going to have a higher risk of not getting good control. But it's much cheaper, much easier to work around some of the regulation. So what fumigant works, I have Telone products there, but it should be Telone and some other products that are listed as well, so pichlors and things like that. But that 1,3-dichloropropene is probably the most common one that's being used in mixes in the southeast that are going to be able to control nematodes. And then we heard some talk earlier about VAPAM, which would be the metam products, metam sodium, potassium. I'm not an expert on fumigants. I've done some work in phosphine if you have questions about that, but I'm just kind of telling you what most people are going to recommend to you. But I would say always consider the biology of the pest you hope to manage. And so if you're only putting fumigant or some kind of fumigant like 10 to 12 inches in the soil, you could ask yourself, are dagger nematodes that they can find easily 24 inches down, are you going to be able to get rid of all of those with just one single fumigation? So probably not, is what I would say. I put up the Jonathan Oliver's guide that he mentioned earlier, the Southeastern Caneberry guide, to show you what's recommended there. You can see Pichlor 60, Pichlor 80, VAPAM, KPAM, very good to good. Then Telone 2 would be excellent. So just really how much of that 1,3-dichloropropene that you're getting in the mix, and then also the KPAM and VAPAM, which are the metam products, are what's recommended. But I really recommend you take some time to read that section. It's towards the beginning of the Southeast Region Caneberry Guide to look at it. I also looked at the Pacific Northwest and kind of what products they're recommending, and they actually have some suggested prices that a lot of people are paying out on the West Coast to have their area fumigated, just to give you some ideas per acre and what they're paying and some of the limitations. Now that's in Oregon, Washington, so those limitations aren't going to be the same that we have here, or potentially they could be, but there may be some more regulation on them. But you can see that, control of nematodes, most of those products are going to get you there, but VAPAM, I don't think they were as confident in as some of the others, which was the same in the Southeast Guide. I've also looked at some research, whether or not fumigation is completely ridding the problem. And I like this paper because they really looked at depth in the soil. So they went 3 bars there. You have the black bars pre-fumigation, then there's white and gray, which is going to be really hard to see, but they're stacked pre, post-fumigation, and then at planting. And so there was some time in between. But this is ripping plants out, then going back in after you go through the dormant period to plant new plants. That's what they're looking at. And this is P. penetrans, so this is going to be your root lesion nematode. And what they found was that even after post-fumigation, they weren't getting rid of all of them, and especially as you went deeper. And this root lesion nematode is actually going to be a bit further up in the soil profile than our dagger nematodes are normally found. So you can expect this effect to probably be a little bit more accentuated in a nematode that's going to be further in the soil. So fumigation is not getting rid of all of them, is kind of what this is showing. And a pest like root lesion nematode, where you're trying to lower the numbers and that's going to get you a long way, that's not such a bad thing. But in dagger nematodes, where you're hoping to not have them at all, it's not exactly lowering your risk as much as you would hope. So when you're thinking of control strategy for nematodes, what you really need to keep in mind is that no single method will rid your farms of nematodes or prevent injury. Fumigation is not a silver bullet. I know a lot of folks are using it, but it doesn't mean you're not going to have the issue afterwards. Now, if you start out with very low numbers, it's much more likely that fumigation is going to do a little bit better of a job for you, because you're going to have a higher risk of missing what's out there. Based on the biology of dagger and root lesion nematodes, I don't know that we should expect that single row fumigation is going to solve your issues. And you can expect in these perennial plants that you're cutting rows sometimes in between where you were last time. There's going to be a high level of inoculum of these nematodes in between where you plant in the coming year on your caneberry crops. And so, especially for the next year, but maybe even for 2 or 3 years later, based on the biology of some of these nematodes. So what else should you be employing with your integrated strategy for managing nematodes? I would say just basic cultural control, crop rotation. Not going from caneberry to caneberry year to year, and I know that's not possible everywhere, but in Arkansas, that's what we try to recommend, and that's what a lot of people are trying to do, is trying not to put caneberries right back into where they were the year before. And I know that it's hard to justify that everywhere, but it is a good approach to try. We know that lesion nematodes can be 8 or more months on dead root material. So if you're planting right back in, there's a high chance that they're going to be able to move right back from the eggs that hatch from that in the soil onto what you plant into the ground. The other thing is there was some work showing that virus-infected roots can actually act as a reservoir for those and actually be fed on by dagger nematodes 10 years or more later, and then spread that virus onto some other crops that you put in the ground. So the more time you're putting in to get rid of these things or leave it a little bit fallow, it's going to give you a lot. Fallow periods after crop destruction. And so when we say fallow, we absolutely mean fallow with especially dagger nematodes. They can feed on a lot of weed species and move these viruses around. Just because it's a tobacco green spot doesn't mean they need tobacco. They can be a lot of different types of weed species, especially solanaceous weeds that are very common in blackberry fields. Repeated tillage of fallow soil. And so Dagger nematodes can be reduced by repeatedly tilling the soil. It's not always going to be the best thing to do to try to help with your soil health and things like that, and you're not going to get the ones that are deep down, but it has been shown to reduce numbers. And then there's some idea of cover crops and soil amendments. So complex soil food webs are generally going to be a harder place for a single species to survive. I don't know why the back half of that sentence got cut off, but the other thing that there's been a lot of research recently on are this idea of cover crops, right? Dagger nematodes may move very far down in the soil, down deep into the soil, but if you put in some shallow-rooted grasses or just get grass cover crops in, they're going to move up to try to feed on those grasses initially, and so that's going to bring them closer to where you would put in a fumigant later on. And so there's been work showing that a shallow-rooted grass cover crop ahead of a fumigant actually increases the efficacy of that fumigant itself. So thinking of cover crops, what is a suitable cover crop? And so it's kind of complex depending on what nematode species you're dealing with. So mustard and other brassica cover crops can suppress pathogens. They have glucosinolates that you can use to try to help kill some of these plant pathogens. But you really don't want to look closely because they may also act as a small reservoir for some of the viruses that are sticking around in a very long-lived dagger nematode species. The research on that's not definitive, so it's not to say that they're not going to be good. But what we do know is that grasses are going to do a really good job on dagger nematodes because there's work showing that once they feed on grasses for extended periods of time, they lose the viruses that they had previously, and also they're going to move further up into that soil structure that you want. And so for lesion nematodes, some grasses may not be great hosts, great non-hosts, I don't know the best way to say that, for lesion nematodes, which means basically you put out something like sorghum Sudan. Some research has shown that they put out sorghum Sudan, the root lesion nematodes didn't really decrease as well as they did on things like, I believe it was oats and some of the rye species, but it was pretty inconclusive. And so it's possible that Sudan or some other things may not be a great thing to get rid of lesion nematodes, but it's going to be a lot better than some of the alternatives like any type of legume which can harbor some of these viruses. Post-planting options, there's very few available, so there's very little data in general supporting management in Blackberry once you have plants in the field. It's very hard to find anything on it, to be honest. In raspberry, they've looked at oxymil, which is vidate. There's a heavy restriction on caneberry species, so don't go out there and spray your blackberries unless you read a label very closely, because my guess is it's not labeled on blackberries, but it may be labeled in some states. Vellum prime, there's been some work in raspberry to show that it's pretty effective, and also in grapes, I believe. And then some biologicals. There's a lot of different options out there that were some suggested potential benefits for. But for dagger nematodes and blackberry, not sure. You know, all the work that I've seen on vellum prime has been on endoparasites, things that are inside of the plant roots. Vellum prime is going to move through the plant some. It's going to contact a lot easier than an ectoparasite like dagger would. So we know that it's not effective against eggs either. And so is it going to really reduce these numbers? I'm not sure. I'm not seeing good work to show whether one way or the other. So I wanted you to give an example of the soil reports we're getting in Arkansas, just to give you an idea of how many nematodes we have. And so there's one here where we have 1,462 dagger nematodes in the soil. What did I tell you, 10 or 120 was our number that's bad? There on the bottom we have lesion 154, dagger 731. So this is what we're getting a lot of questions about this because we're seeing so much. And so just real quickly, I'll show you some data from a nematicide trial that we did this year or this last summer. And so we had a grower that had two different fields we looked at, and so we ended up going with that field 46. And so we see we had about 100, or between about 850 or so dagger nematodes across the field pretty consistently. And then we had somewhere around 150 to 200 or so lesions. This was a soil test, I believe, in late February, early March. And so the lesions definitely increased, which is what they also see in Pacific Northwest. But we just wanted to see how this organic nematicide did, and then also compare that to vellum prime, because we didn't have any data at all on blackberries on that. So you can see our untreated there. And we applied it twice. We did two different rates of the organic, and then we did the high rate of vellum, which is what they recommend. So you can see there the field. We had 2 plots per row, so it was a 4. Across 2 rows, we had one set of 4 replication, or one set of a replication, then we had 4 total. We put it out with a single nozzle boom at about 100 gallons per acre. It was right around a rainfall, so we were pretty confident it went into the soil on just a 24-inch band. There was no plastic or anything to prevent it. And then we sampled 8 cores from each plot and made a mixture of that and sent it into our nematode lab to get counted. We sampled at prior to the application, 28 days after, and then 60 days after, and we applied around end of March, and then again a month later in April, which is about what you would see recommended for a raspberry or something similar. So here's our data. You can see number of dagger nematodes on the left there, and then our different treatments on the bottom. And within each treatment, you can see our pre, mid, and post samples. So before we sprayed, a month after, and then 2 months after the first spray or a month after the second one. And for dagger nematodes, I hope you can find something there, but we couldn't. So we didn't see any good effect on dagger. It seemed like it was all over the place, the same in every single treatment. Not really anything going on. We did see something that was a little hopeful for root lesion, which is what they've seen in raspberry as well. And so you can see the untreated there where initially it was a little high and then it kind of went down, but Velum Prom was really the only one where we saw this nice decrease within the treatments where it actually decreased the population significantly. And so we're hopeful that this would be an option if someone had a lot of root lesions. So that's 300 to 400, maybe a little under what they would say in raspberry was a problem, but we think it's an option someone could use. So we really not really too hopeful on the biological nematicide, but we haven't done enough work, I would say, to truly give it a good shot. And so it didn't have any impact on the dagger root lesion nematodes that we saw, but I don't think you could ever look at those things 1 year and decide anything about it, because it's about trying to apply it over time and build up some type of soil health or something like that to make it a less hospitable area for dagger nematodes. But I'm not sure if it would work or not. Velum Prom did not reduce daggers, but maybe it would be a little better if we tried it with the drip application. It's kind of hard to do that on on-farm research, but we think it's a valuable tool for suppressing root lesion nematodes, and that kind of runs similar to what we've seen in other regions and other caneberry crops. So with that, thanks to Ryan and Jenny and Jess, our interns that helped, and Keela, our graduate student. I forgot to put her name on there, but she did a lot of the data collection of this on the back end. And then the growers should keep letting us do on-farm research. We're very appreciative of that. But these are some of the resources that we have at the University of Arkansas, or also at the smallfruits.org, the Southern Region Small Fruit Consortium. So if you definitely try to access those if you can, and if you have any questions, I'd be happy to answer either now, or you can holler at me on my cell number or my email. So thanks. And I think we have time for like 2 questions if somebody really wants to put me on the fire about nematodes, make me uncomfortable. Yep.
Some of those high levels of nematodes, like you said, in some areas like 10 dagger, it's like that, right? But then you're getting numbers that are 1,500, 1,400. So do you feel like the damage really comes from the lack of biodiversity in the soil, or soil type, or like what makes the difference between a 10 dagger that's going to kill everything versus a 1,500 that you can still grow dry?
Yeah, I would say dagger, you're not going to see a direct impact right away, and so I think it's a slow burn. What we've seen is that where we don't have it, we don't see it just show up out of the blue, and so I think this is just a facet of continually growing caneberry in the same spots year after year, and so it could be moved around some on equipment from other plantings on the farm, and that's how it could get going. But I think it's just a slow increase in numbers, and I think in a blackberry soil, that ecosystem is going to be very friendly to these nematodes. So I don't know that you're going to really get out of that by doing anything. I don't think you're going to add something that's going to make it so unfriendly these nematodes aren't going to do well. It's just a facet of agriculture, in my opinion. But I think it's just risk of virus transmission. And I'll tell you that this field had a lot of virus in it. And it was I think 3-year-old or 3 or 4-year-old Ponca field, and it produced almost nothing, and probably a lot due to the disease and also the lesions, nematodes that were there. Any other questions? Okay. All right, Wayne. Oh, yeah, we got Wayne in here. So next up, we have Wayne Mitchum, NC State weed specialist and also blackberry grower, so you can ask him from both sides of his hat on, hat off kind of deal.
All right, I'm going to talk to you about weed control in blackberries. Weeds compete with everything for water and nutrients, and of course, if they get tall enough, they're going to compete for sunlight. The big thing with starting with a blackberry weed control program is, especially in the establishment, you've got to start clean. You can't start out with an area and be planting in an area that's got some small weeds emerged. Early is always better than late with applications. Timing on this is essentially everything. And weed management programs, it's very important that you plan before you plant. I think if you've got problems in a site, if you've got nut sedge, for example, that you're going to be planting into a site, there are ways you can deal with that ahead of time, or even something like Bermuda grass for that matter. There are some things you can do prior to planting, like high rates of glyphosate, maybe even the fall before you plant the fall and the spring can be very helpful in reducing that pressure the following year. Keep new plantings as weed-free as possible. We want to maximize that growth. That first year, the more growth we can get in that first year, that means we're going to have more production the following year when we actually go and begin to harvest. The one thing about weed management in established situations is I think we have to keep in mind that weed management starts after harvest for the following year, and we have to have that mindset in order to do a good job of keeping those clean. Spring and summer weed control can be very important to us. And there's going to be a slide here in a moment where I'll mention this even more so. But the last week of February through the first week of March in the Piedmont of western North Carolina, the upstate of South Carolina, middle Georgia, that would be a critical time in which you need to make sure you do a good job then of eliminating any weeds that are up. Because if you miss that application timing, and if there's anything emerged that you don't take care of at that time, or they get too big, you're going to have a problem the rest of the spring and going into the summer. We need to ask the question, if you think about your fields and your blackberry plantings, how weedy are they now? And that's going to give you a thought about, when I need to be doing things and when I need to be taking care of business as we go into spring. We're going to start with going over newly planted situations. A lot of folks like to use black plastic mulch as a cover. There are some benefits to that. I do think that, that first year it's definitely a lot easier to control the weeds. You essentially have to control the weeds around the holes and on the edges of the beds. So that makes it very nice. I think long term, though, there is some real setbacks to that. And there's some other issues that I think are a little bit of concern. You have to have a little benefit from rainfall because it is a black plastic cover, and you're going to have to depend a little more on irrigation. If you've got fields with slope and you've got low and high places, you're going to have basically issues of drainage and water standing and moving around that bed. But the weed control is a lot better in that first year. But when you get into the second year, you got into questions of what do we do with the holes in the plastic? Because as you go into harvest, they're going to be putting feet on those. Your workers will be putting their feet on those, picking. Just inevitable, you're going to get holes in the plastic, and then weeds are going to come through those. And if you just want to get really honest about the situation from a herbicide standpoint, we don't have any herbicides labeled for use for caneberries growing on black plastic. I mean, if you look at tomatoes, you've got tomato production on bare ground, and you've got tomato production grown in black plastic, on black plastic. Look at strawberries. We've got perennial systems, we've got annual systems. And technically, really don't have anything labeled for use in blackberries for herbicides grown on black plastic. That being said, the truth of the matter is we growers are banding some stuff besides the bed, and there are some things you can do. But if you really want to get down to the brass tacks of it, it's probably not going to be looked at as technically a labeled situation. With bare ground, the weed control there is obviously cultivation and herbicides. As we move into that, weed management in newly planted blackberry, for a long time we depended a lot on surflin, which is Horizon, or generic products. That's no longer available. It's not going to be available to my knowledge. But Satellite Hydrocap is the only labeled alternative to that. It is a pendimethalin product. It contains active ingredient of pendimethalin, and that's the one you would need to use as an alternative to Surflan or Horizon. I do think it's one option that we have to think about, and one that I've started using when we've planted some blackberries on the farm recently, is using wax-coated beverage cartons as a means of protecting those young plants. We place those over the plants. You can do it after transplanting, but what ideally I like to do is I like to get those things planted, get them off and growing, and then go back and put that carton over and use a piece of bamboo like you use in grapevines, cut that in about a 12, 14-inch length and put it down beside the plant to hold that milk carton in place, and then pull a little soil around it to keep it from moving when you have wind. The nice thing about that is it does give you some protection. When you direct herbicides, we can use gramoxone a little bit earlier than we would typically be able to if we didn't have those protected. Injury can still occur if you spray when it's too windy or you got the incorrect nozzle height or using high pressures, you can get drift. So you have to, it's not a total prevention of all things, but it does help out quite a bit. In newly planted situations, we're on bare ground. I like the idea of planting those young plants. You're going to have your irrigation system out there. You can go through with a cultivation, and I really like doing that. We don't have enough root expansion from that newly planted plant that you're going to cause any damage to those roots as long as you do it when they're fairly small. But then once those plants go to getting 4 or 5 inches of new growth and beginning to get some height to them, go in and put your milk cartons in place, and then you can go in and direct the application of a half rate of simazine with 2 or 3 quarts of satellite hydrocap for residual control. Once that residual control breaks down, hopefully that's going to give you 60 days or so of good pre-emergence weed control. But if you need to, you can go back then with paraquat as a directed application. We can use clethodim or some other graminicides to take care of perennial grasses if we needed to. But I just think that's a nice program in a bare ground situation that works fairly effective and does a nice job for you. In established blackberries, we've got a lot of options, and there's a number of different herbicides registered. What I'm going to talk about here is things that I like the best. Going in post-harvest, going in that after you've removed those fluorocanes, putting out a directed application anytime up until October of simazine at 2 quarts plus rim at 4 ounces, a rim sulfuron product, which would be like Matrix, with paraquat is one option. The Matrix products with satellite hydrocap would be another option, and then the Chateau at 6 ounces would be an option. Of course, there's a number of generic Chateau products available and flumioxazin products that would be alternatives to that. And the idea with that is to sort of clean up that those weeds that are there after you've removed those primocanes. Try to give you some residual weed control into the fall, and because we will get some new growth, new flush of growth in the fall on those primocanes, and we're going to be trying to grow those for that crop the following year. So I just think that's a good idea to make that application and put that into place. And then here's the application, though, that's extremely critical, in my opinion, to good success for weed control through the spring and through harvest. And that's going to start in February or early March, like I said. I live between Charlotte and Asheville. That's where our farm's at, directly north of Gaffney, South Carolina, about probably 35 miles. And the first week of March is a good time to start. We want to make that application just before those winter annual weeds begin to explode and take off as things warm up in the spring. And what we found is one program that I have used and been sort of my standby is that send bar at 1/4 to 3/4 of a pound. Of course, the heavier soil you got, the higher rate you can use. If you're on a sandier soil, you'd want to stay down at that 1/4 to 1/2 pound. With 4 ounces of a Matrix product and paraquat. That works really, really good. And we get good post-emergence control of some difficult to control weeds. For example, one would be horseweed. Horseweed is not controlled by paraquat, but if we put that Sinbar and rim sulfuron together, we get very nice post-emergence horseweed control, so long as that horseweed's not over 2.5, 3 inches tall. If we wait 2 weeks later and that thing's 4 inches tall, it's going to be 4 4 foot tall before you can do anything about it. I mean, that's just the reality. But the other options would be use Clisto at 3 to 6 fluid ounces with RIM or a matrix product. Allion is a very good pre-emergence herbicide. Doesn't have any post-emergence effect at all. We're sort of restricted on when we can use it. It's got to be kind of in a dormant application. But 3.5 to 5 fluid ounces of Allion with some RIM does a great job with paraquat. We need the RIM in there for some extra post-emergence activity, for example, on something like horseweed. Chateau and RIM, another good option there to give us good residual control with the paraquat. Again, it's extremely important that you make this application timely. Those weeds need to be ideally less than 3 inches tall when you make this application if they're and have good success with a post-emergence control. If we don't kill what's up, there is going to be a problem the rest of the end of the spring, rest of the time into the spring. Prickly lettuce and henbit can also be a problem for us as well. Then when we get closer to primocane emergence, and hopefully if you've made that application, if you've been timely and you got good post-emergence control, as we go into the primocane emergence, which is going to depend a little bit on how fast the soil warms up in the spring, but typically that's going to be for us somewhere probably around the last of April, the 1st of May where I'm at. And I like to go back in with another pre-emergence herbicide application then. And, if you've done a good job post-emergence, you may or may not need paraquat with that. It just depends on how clean and how good a job you did earlier. But typically, the ground's going to be pretty clean from that initial application you made back in the February, March time frame. And depending on what you use then, we'll make a decision as to what you can use this time. If you've already used Chateau in February, obviously we can't use the Chateau application again. But the Chateau at 6 ounces of RIM is an option at this time frame. Zeus Prime at 10 to 12 fluid ounces with Cytolite Hydrocap is a great pre-emergence tank mix. The nice thing about Zeus Prime is it's sort of like Simazine on steroids. You get more activity. The nice thing is if you have nutsedge, it's a very good nutsedge herbicide pre-emergence wise. So it does a really nice job, good residual activity, and that's one that probably needs to be utilized a little more than it is. And it's got labels not only in caneberries, but it also has great labels in apples and grapes. And then Simazine with Satellite Hydrocap would be an option. And then if you've got a heavier soil, a pretty clay soil, Sinbart 1 to 1.5 pound with 4 ounces of RIM would do a really good job as well from a pre-emergent standpoint. And of course we could include paraquat with those if we need to do that. The only thing I would say about the Sinbart 1 to 1.5 pound is we have to be careful using that year after year. We can see some Sinbar build up a little bit over time. Typically it'll show up on the ends of the rows first where you slow down to make turns, but it can cause some yellowing and some stunning if we use that over and over again, and over time it builds up. A good herbicide, I wouldn't be afraid of it. I just think you got to be wise in how you use it. And of course with perennial grasses, we'd have to deal with those. Fusilade in a bearing situation is what we'd want to go with because it's the one that has the 1-day PHI, and that's going to do what we'd use it there for would be, of course, Bermuda grass, Johnson grass, those type weeds. The other products work really well, the Post and the Select Max, but the reason we go with the Fusilade is, again, because of that short pre-harvest interval. Select Max has a 7-day PHI. Post has a 45-day PHI. And we know that in order to do a good job of controlling Bermuda grass or Johnson grass for that matter, we have to make an initial application when Bermuda grass, for example, is about has 4 to 8 inches of new growth in the spring. Well, that's going to be somewhere in late April, say, maybe early May. And then we're going to need to follow up with a second application when we get regrowth from that initial application. Well, that could be, 4 weeks or so after that. And if you go to looking at PHIs for Post, we're not going to have enough time to get that second application on and not be within that 45-day post-harvest interval, or we would be well within that, I should say. And so that's why Fusarium is the one I kind of like to go to there because of that. Couple new options. Break On is a relatively new product. It's an old herbicide with fluoridone. It's been around since I think the 70s. Kind of been brought back to the forefront in cotton here several years ago now. And because it was a good residual product to manage glyphosate-resistant pigweed in cotton. And it does have some fruit uses. We've got an apple label and a peach label. The use rates on that's 21 to 43 fluid ounces per acre. We can use it on blackberries that are established at least 1 year. We cannot, the label says do not use it more than 2 consecutive years in a row. So if you use it 2 years, you definitely need to take a year off. Of course, it needs rainfall to activate it. Doesn't have any post-emergence activity, and it has worked well in tank mixes with other pre-emergence herbicides like rimsulfuron, which is Matrix, Sinbar, and Chateau, or flumioxazin products. You know, it's pretty broad spectrum, broadleaf and grass weeds, has a 30-day PHI. This is some work we did where we did some sequential applications. That was 21 ounces this was in apples actually, but 21 ounces like in March, followed by another 21 ounces there in June, which and obviously with blackberries, that application couldn't be made that late, but we could make it in that February time frame and follow it up maybe in that early May or late April time frame, depending on the variety you've got. But the break with 21 ounces by itself did a pretty good job, but it wasn't quite as good as when we put some matrix with that. And when we put the matrix with it, it did a really nice job. And that was actually only a half rate of matrix. So we can use as much as 4 ounces of matrix with the break on. So it's just another option you have, and one that has done a fairly good job. Another post-emergence option that's new or relatively new is Quinstar. Quinstar has quinclorac in it. That's a rice herbicide. It's got some sort of phenoxy type activity, but it does have activity on grasses as well as some other broadleaf weeds. It's pretty good on clovers. You know, for the folks in western North Carolina and up in the mountains, they have some issues with field bindweed. It happens to be pretty good on that. It's pretty good post-emergence on morning glory. It's pretty good post-emergence on prickly lettuce, which could be a problem in that winter March time frame, early or late February time frame. So just as another option for you that you could think about incorporating into your program. You know, woody perennials are no question a problem, and you know, we generally get by without a lot of woody perennials that first 3 or 4 years, but over time, you know, things like horse nettle and Virginia creeper and even poison ivy and pokeweed all become problematic. And the reality is the things that control those weeds tend to control blackberries. And there's just not a real good option for us on those at this time currently. You know, we can spot treat with glyphosate, but you have to realize that when you do that, you're probably going to sacrifice or damage the caneberries that are close to that where you're making those applications. And, you know, 1.5%, 2% solution of glyphosate works pretty good on Virginia creeper as a spot application in late summer and fall. Pretty much the same thing for poison ivy. You know, pokeweed, it's a little bit easier to control with a spot spray when it just comes up in the spring. So that would be when you would take care of that. Horse nettle is most susceptible to glyphosate when it has flower buds. So they're all a little bit of a range here on when you'd make these applications, and it's just a hard problem to deal with those weeds because of the fact that they are very difficult to control, and our herbicide options are not that good. And physical removal is another option. You know, something like Virginia creeper, you know, when you see it first come up and you find it, you probably need to go ahead and take care of it, because Virginia creeper that is, you know, about like this thermos is to now, next year it'll be the whole length of the table. So we have to take care of those pretty quickly. You know, future post-emergence herbicide registrations, we're looking at Sandbar now in newly planted situations, and that would be another option for, getting a planting established. I think probably the more exciting things that's coming are some options we're going to have here post-emergence to manage some of these weeds. It's going to help out on some of the problematic weeds. Embed Extra is the 2,4-D choline that they're using over the top of 2,4-D tolerant crops. We've done work with it several years ago. That's currently going through a registration process. The nice thing about that is, it's gonna knock back things like horse nettle. It's not gonna be a magic silver bullet by any means, but it's gonna be way better than anything else that we have. It's gonna work on pokeweed. It will give you pretty good knockdown of Virginia creeper. Be great on horseweed, Carolina geranium, prickly lettuce, and thistle. Trevix is another product that we've looked at. It's a PPO inhibitor. We've got a product called AIM that's basically labeled for post-emergence use in caneberries. But Trevix is a much more effective post-emergence herbicide than AIM. It's a lot better, I think, on horseweed. It just does a better job. I've seen it do a really nice job on prickly lettuce post-emergence, but it would be a nice option to have to put in application there as well. And I could really see where Trevix would work well, in that winter time frame. If we had the option to put out Embed and Trevix together, that would be a nice alternative to paraquat. To give you some pretty broad spectrum knockdown of those winter annual weeds. And if we put something like Rimsulfuron with it as well, I think we could do a really nice job post-emergence there and not have to use paraquat. Glufosinate is another one that they're working on getting labeled. Probably one I'm like the idea of having glufosinate. I think it will be a nice addition because it's another non-selective herbicide. That's an alternative to paraquat. The only thing I'm a little bit worried about is, and we've talked with IR4, and I'm sure they've talked back to the manufacturer about getting this thing labeled where we can use it correctly. That timing on that, it needs to be done before primocane emergence or after harvest, because it is very, it's not systemic, but it's locally systemic. I mean, you can use it for sucker control on grapes and other things. And actually, if you look at some of the older labels, it listed use for caneberry or blackberry control as a weed. So I think we need to just be careful how we use it. And I've looked at it on our farm and done some replicated trials. Haven't seen any damage, but if you put it out on primocanes, it's going to work on those pretty hard. So I just think that's going to be the key. We need to know when to use it and the placement of that application timings. And the fact is, those would be when we need it most anyway, would be in that spring, early spring time frame before primocane emergence or going in post-harvest where it would have its greatest fit for us anyway. But, and when I go to thinking about glufosinate, I really like the idea of putting it and 2,4-D together. Talk about a broad spectrum, non-selective post-emergence burn down. That would be pretty nice to have. We've done some work with that in apples and peaches, and we've had as good a results with 2,4-D and glufosinate together as we have with glyphosate and 2,4-D together. So we can't use glyphosate really in caneberries because of the injury potential, so we wouldn't want to do that. But that combination of the glufosinate and the embed would give us something that would be really, really good and far better than anything we have right now. That concludes my presentation. That's me and the family. And if you've got any questions, I'll try to answer them. Yes. we used it, we did some work with it this past year and the year before, and I haven't seen any problems with it, and was it light textured soils or?
About medium.
Okay. We haven't seen any problem with it. And sometimes I do wonder how good folks are at knowing how much material they're really putting out. And that comes up from time to time. I get questions all the time of, well, how many ounces do I need to put in 20 gallon? Well, that ain't the way this deal works. So if you're seeing a problem like that, I would probably, first thing I'd do is go back and look at the calibration deal and just make sure he's putting out what he thinks he's putting out. Because I do think that is a problem. And I do think sometimes people, if they're not a traditional ag person, they may be concentrating those rates. Even though it's a banded application, they may be thinking concentrated rates, and I worry about that sometimes. So I don't know the background of the person you're dealing with, but that's something you have to think about. But we hadn't seen any problem with it when we've put it out in our trials.
Do you like Allion?
Oh man, I love Allion. I really don't know why the label restricts it like it does. I don't know why they have the dormant application window on that, but it's a great residual, a lot of residual control. As far as herbicides with residual control, it's the best, most residual you're going to get out of anything. Morning glories can be a little bit of a weakness for it. So a real high morning glory pressure, you might need to put a little something else with it. When we put other things with it, it and rim sulfuron works really well. Helena's got a premix called Centris. That's a premixture of the Allion and the rim together. But those two work really together. Great pre-emergence herbicide. And the other one I really like a lot is the Zeus Prime. I think that's one that probably needs to be utilized a little more. But it does need something with it for residual grass control. Anything else? Yes.
I learned a lot of the weed control, and I appreciate that information. And I just have a question. You are focused on the weed control in the caneberries and field and farmland, and what is actually treating only farmlands, or are you treating wherever you want? My question is, certain weed controls, especially the timing is important.
Yes. So you're worried about shortage of?
No, I'm just talking about your idea is just control the trails of.
Yeah, well, what we typically do is we're just controlling about a 6 foot wide herbicide strip, 3 foot on each side of the road. Yeah, and where I'm at, we leave grass in the middles because of erosion and that sort of thing. Now you get in South Georgia and the coastal plain of North Carolina, fescue doesn't do as well. They typically have mowed native vegetation. But we're just banding those. And, we pretty much feel comfortable about a 5 to 6 foot wide herbicide strip is probably where you need to be with caneberries. Anything else? All right, thank you.